Far below the surface of the Pacific Ocean, there are creatures alive right now that should not exist. At depths where sunlight has never reached, where the pressure would crush a human body instantly, life did not just survive. It became stranger than anything evolution produced on land. A fish was discovered more than 8,000 meters down, with flesh so saturated in chemical stabilizers that scientists describe the taste as ammonia. Yet down there, it is an apex predator. A jellyfish no larger than a fingernail has been observed reversing its own life cycle, transforming from adulthood back into youth again and again, with no known biological limit. In 1977, scientists lowered cameras into the deep ocean expecting barren rock and found entire ecosystems thriving without sunlight at all, rewriting one of the most fundamental assumptions in biology in a single expedition. Some of these creatures live beside hydrothermal vents, releasing water hot enough to melt lead, while the surrounding ocean remains near freezing. Some have eyes larger than a human head, others have no eyes whatsoever, yet move through complete darkness with impossible precision. And at the deepest surveyed point on Earth, soft-bodied animals live permanently beneath pressures strong enough to collapse steel equipment. These creatures did not adapt despite the deep sea. They were built by it. So how does life continue in a place that seems designed to prevent it entirely? And what does it mean that the largest living environment on Earth has remained hidden beneath us this whole time? Today, we descend into the deepest parts of the ocean to uncover the creatures that broke every rule biology thought it understood. If you enjoy this kind of content, a like and subscribe helps more people find it. And I'm curious, what brought you here tonight? Can't sleep, or just love this kind of thing? Let me know in the comments. Now, let's descend into the darkest living world on Earth. Far below the movement of storms and tides, the ocean separates itself into layers so vast and isolated they resemble stacked worlds more than parts of a single sea. The uppermost region, the sunlight zone, extends from the surface down to roughly 200 meters, the final boundary where sunlight still carries enough energy for photosynthesis. Beneath it lies the twilight zone, descending toward 1,000 meters, where daylight weakens into a dim blue haze before vanishing entirely. Below that begins the midnight zone, stretching through 4,000 meters of depth in uninterrupted blackness. Farther still rests the abyssal zone, a plane of immense cold between 4,000 and 6,000 meters. Beyond even that lies the Hadal zone, the trenches, descending deeper than 6,000 meters into fractures in the planet itself, places so remote that they remained almost untouched throughout the entirety of human history. And yet the strange reality is not simply how deep these places are, but how little of them humanity has truly seen. Roughly 23% of the ocean floor has been mapped in meaningful detail. More complete maps exist for the surface of the moon than for the seafloor beneath Earth's own oceans. Most of the planet remains hidden underwater, no human eye has witnessed directly, concealed beneath pressure, darkness, and distance, so extreme that exploration becomes painfully slow and enormously expensive. The scale of the deep sea reshapes the meaning of the word environment. Everything below 200 meters covers approximately 65% of Earth's surface and represents almost 95% of all living space available on the planet by volume. Forests, deserts, mountains, and cities exist only within a thin surface skin compared to the immense vertical wilderness below. Most life on Earth does not live where sunlight reaches. Most of Earth's habitable space exists in darkness. Reaching it demands machines built more like spacecraft than boats. At great depth, water presses inward with enough force to collapse ordinary steel structures within moments. Submersibles designed for the abyss require thick titanium hulls engineered with extraordinary precision, each descent carrying risks severe enough that a single dive may cost more than a space shuttle launch once did. Every cable, seal, camera housing, and robotic joint must survive an environment actively attempting to compress it into ruin. Even temperature becomes an obstacle to life. Beneath roughly 1,000 meters, ocean water across most of the planet remains between 2 and 4 degrees Celsius almost continuously. In laboratories on the surface, temperatures this low slow biological activity dramatically. Many familiar processes begin to fail, muscles weaken, chemical reactions lose efficiency, membranes stiffen. Yet the deep sea exists in this condition permanently, cold not as a season but as a stable planetary state. Food drifts downward slowly from the world above. Without sunlight, photosynthesis disappears almost entirely, and with it vanish forests, algae fields, and nearly all plant life. Most deep-sea organisms survive on what marine biologists call marine snow, a constant but sparse fall of dead plankton, fragments of tissue, fecal particles, dust, mucus, and dissolved organic debris descending from surface waters over days or weeks. By the time it reaches the abyss, little nutritional value remains. Life persists anyway. Darkness there is not comparable to night on land. Night on the surface still carries moonlight, starlight, distant illumination from cities and clouds. The deep sea has none of these. In many regions, no natural external light has existed for millions of years. The only illumination comes from living organisms themselves, pulses of blue and green bioluminescence appearing briefly in water, otherwise untouched by light, since long before human beings existed. Before 1977, biology interpreted these conditions with confidence. Scientists expected the deep ocean below the photic zone to contain little life, low diversity, and ecosystems fully explainable through familiar rules of energy and survival. Extreme pressure, permanent cold, starvation, and darkness appeared too severe for anything else. The abyss was imagined as biologically sparse, perhaps even approaching lifelessness. One expedition after another would eventually lower cameras into that darkness and discover the opposite. In February of 1977, a camera descended through the Pacific Ocean toward a section of fractured seafloor known as the Galapagos Rift. Nearly 2.5 kilometers beneath the surface, the water had long since passed beyond every familiar sign of the living world above. No sunlight had reached those depths in geological ages. The descent moved through cold layers of open water until even the faintest traces of blue disappeared, leaving only drifting particles crossing the camera lights like ash in darkness. The scientists aboard the research vessel Nor were not searching for animals. The expedition had been organized around geology, not biology. Their goal was to examine hydrothermal activity along the ocean floor using a towed camera sled called Angus, a machine built to photograph volcanic fractures and mineral formations hidden beneath the Pacific. They expected warm water venting from cracks in the crust, perhaps unusual rock chemistry, perhaps mineral deposits forming around underwater volcanic systems. The deep sea itself was still widely imagined as sparse and quiet, a place where survival narrowed toward near absence. Then the seafloor appeared. Around the vent openings stood dense white structures rising from the darkness in clusters, some taller than a human being. Giant tube worms swayed in the moving water beside clams the size of dinner plates. Crabs crossed fields of mineral deposits. Shrimp drifted through shimmering plumes of heated water. Fish circled slowly through an environment no one had believed should contain enough energy to sustain such abundance. Instead of isolation, the camera found community. Instead of scarcity, it found concentration. The volcanic fractures were surrounded by entire ecosystems, layered together with the density of reefs or forests. The immediate scientific problem was simple and severe. Nothing in that environment had access to sunlight. Since the foundations of modern biology, the sun had been treated as the beginning of every complex food chain on Earth. Plants transformed solar energy through photosynthesis. Animals consumed plants or consumed other animals that had eaten plants. Even in the deep ocean, life was understood as ultimately dependent on material drifting downward from illuminated surface waters. But these organisms existed too far below the reach of that system. Their world appeared disconnected from the surface entirely. What the scientists slowly realized was that the vents themselves were feeding the ecosystem. Hydrothermal vents release hydrogen sulfide from beneath the Earth's crust, a chemical toxic to most surface organisms. Around the vent openings, bacteria were using that chemical the way plants use sunlight, extracting energy from it through a process called chemosynthesis. Those bacteria became the foundation of an entirely separate biological chain. Small organisms fed on the bacteria. Larger organisms fed on them in turn. Life was flourishing around volcanic chemistry alone, without a single photon of sunlight entering the system. The discovery altered biology at its foundation. For generations, science had treated sunlight not simply as a major energy source, but as the unavoidable beginning of all complex ecosystems. The Galapagos Rift proved otherwise. An entirely independent pathway existed, one capable of sustaining dense animal communities in permanent darkness beneath crushing pressure and near freezing water. Among the scientists reflecting on the discovery was Holger Janasch of the Woods Hole Oceanographic Institution, who described it as the most significant biological discovery of the 20th century. The statement carried little exaggeration. The vent communities did not merely add new species to the tree of life, they expanded the conditions under which life itself could exist. The tube worms became the clearest symbol of that expansion. Riftia pachyptala grew over 2 meters tall, yet possessed no mouth, no stomach, no digestive tract of any kind. Inside their bodies lived symbiotic bacteria that processed the vent chemicals directly, feeding the worms from within. The animal functioned less like a conventional organism than a living chamber built around another form of life. As the implications widened, the discovery reached far beyond Earth's oceans. If biology could anchor itself to chemistry instead of sunlight, then worlds once dismissed as sterile began to appear newly possible. Beneath the frozen crusts of distant moons, inside buried oceans sealed away from stars, the conditions for life no longer seemed impossible, only unfamiliar. Along the walls of hydrothermal vent fields, where mineral clouds drift upward through heated water, pale towers rise from the seafloor in dense clusters. At first glance, they resemble mineral formations, thin white pipes anchored into volcanic stone, motionless except for the soft red plumes extending from their tips. But the structures are alive. Inside each tube rests a giant worm called Riftia pachyptala, an organism found nowhere on Earth except the hydrothermal vent systems of the Pacific Ocean. Some individuals grow longer than 2 meters, their bodies hidden within rigid tubes formed from chitin secreted directly from their own chemistry. The tube is not borrowed from the environment like a shell collected by a hermit crab. The worm constructs it gradually around itself, extending upward as it grows, creating a protective pillar against the surrounding currents and vent turbulence. Hundreds of these tubes standing together form something close to an underwater forest, pale columns rooted in volcanic stone beneath the ocean floor. Yet the deeper strangeness lies inside the animal itself. Riftia possesses no mouth, no stomach, no intestines, no anus. No digestive system of any kind exists within its anatomy. By conventional biological reasoning, the animal should not survive beyond the earliest stages of development. Animals consume food, process nutrients internally, and expel waste. This sequence appears so fundamental that it once seemed inseparable from the definition of animal life itself. Riftia abandoned the entire arrangement. Most of the worm's internal cavity is occupied instead by an organ called the trophosome, a dense tissue chamber filled with chemosynthetic bacteria. Within a single gram of trophosome tissue live roughly 285 billion microbial cells. The worm carries them the way forests carry roots or coral reefs carry algae, not as passengers, but as integrated parts of its existence. The bacteria process the vent chemicals and produce organic compounds that nourish the animal directly. Riftia does not hunt, graze, filter feed, or scavenge. It cultivates an ecosystem inside its own body. The red plume extending from the top of the tube functions as the worm's interface with the surrounding water. Through this feathery structure, Riftia absorbs oxygen and hydrogen sulfide released by the vents. Both substances enter the bloodstream together and travel inward toward the trophosome, where the bacteria transform them into usable organic material. The worm then absorbs the nutrients generated by its internal symbionts, surviving entirely on chemistry gathered from the volcanic environment around it. Its blood carries another impossibility. Riftia uses hemoglobin, the same oxygen-transporting molecule present in human blood, giving the plume its deep crimson color. But ordinary hemoglobin cannot safely bind hydrogen sulfide. Sulfide poisons oxygen transport systems in most animals by interfering with cellular respiration. Riftia evolved a modified hemoglobin capable of carrying both oxygen and hydrogen sulfide simultaneously without destroying its own chemistry. The molecule performs a balancing act that standard vertebrate biology treats as incompatible. Even its growth behaves strangely. Riftia is the fastest growing marine invertebrate ever documented, capable of extending roughly 85 centimeters in a single year despite lacking any conventional feeding mechanism. Around the vents, where geological instability may erase entire ecosystems within decades, rapid growth allows the worms to establish towering colonies before volcanic conditions shift again. Some individuals may persist for more than 170 years. Their larvae begin as microscopic drifters released into open water, carried through the dark ocean until they encounter the faint chemical signature of a hydrothermal vent. Upon detecting those compounds, they settle almost immediately and begin constructing their tubes within hours, anchoring themselves to one of the harshest environments on the planet. The longer biologists studied Riftia pachyptala, the more unstable familiar definitions became. The worm dissolved boundaries once treated as fixed. Feeding no longer required eating. Digestion no longer required organs. Survival no longer depended on consuming plants or animals directly. The body itself became something more flexible than expected, less an isolated creature than a collaboration between different forms of life, joined together so completely that separating them no longer made biological sense. There are moments in biology when scale becomes strangely irrelevant, when one of the most destabilizing discoveries in the living world arrives not inside a giant predator or an ancient leviathan, but inside a creature smaller than a fingernail, drifting almost unnoticed through coastal water. Turritopsis dohrnii, commonly called the immortal jellyfish, measures only a few millimeters across. Its translucent bell pulses softly through the sea with the simplicity of ordinary jellyfish found throughout the world's oceans. Originally native to the Mediterranean Sea, it has now spread through global shipping routes into waters across the planet, carried invisibly in ballast systems and currents, multiplying quietly beyond the regions where it was first observed. Nothing about its appearance suggests what it can do. Most animals move through life in one direction. Cells specialize, bodies mature, tissues accumulate damage, and age advances with irreversible momentum. In biology, development has long been understood as a one-way process. An organism is born, grows into adulthood, declines, and dies. Turritopsis dohrnii disrupted that sequence completely. After reaching full sexual maturity, the jellyfish can reverse itself back into youth when stressed by starvation, injury, illness, or environmental instability. Its cells begin abandoning their existing identities. Mature tissue does not simply heal. Instead, the organism enters a process known as transdifferentiation, where one fully developed cell type transforms directly into another. Muscle cells become different tissues. Specialized structures dissolve and reorganize. The jellyfish collapses inward, settles onto a surface, and rebuilds itself into its earliest developmental stage, a tiny polyp attached to the substrate, like a juvenile colony, beginning life for the first time. From that stage, it grows again. Then, under sufficient stress, it may repeat the cycle once more. No confirmed upper limit to these reversions has ever been established. Turritopsis dohrnii remains the only known animal capable of returning entirely to youth after sexual maturity, while retaining the ability to repeat the process indefinitely. Other organisms possess limited forms of regeneration, replacing damaged limbs or repairing tissue, but this jellyfish reconstructs its entire life cycle from within itself, using its own cells as raw material for biological renewal. The implications unsettled assumptions that had once seemed foundational. Aging in multicellular organisms is generally understood as the accumulation of irreversible cellular damage. DNA degrades slowly over time, proteins misfold, mutations accumulate, cellular machinery loses precision. Eventually, the organism reaches a threshold beyond repair. Yet Turritopsis dohrnii appears capable of interrupting that progression, reorganizing its tissues before the damage becomes terminal. Even now, the precise molecular mechanisms remain incompletely understood. The species itself was first described in 1883, drifting quietly through marine catalogs for more than a century without revealing its deeper anomaly. Only in 1996 did Italian marine biologist Stefano Pirano document the reversion process directly, observing mature jellyfish transforming back into juvenile polyps instead of dying. Since then, researchers including Shin Kubota at Kyoto University have maintained laboratory colonies through repeated cycles of reversal and regrowth. One observed specimen completed the transformation more than 10 separate times under controlled conditions, moving backward through its own development again and again. Outside laboratory walls, the process offers no guarantee of endless survival. Most immortal jellyfish still die in ordinary ways. Predators consume them, disease interrupts them, environmental shifts destroy fragile colonies before reversion can occur. Their immortality exists not as invulnerability, but as biological potential, an escape route embedded within the architecture of the organism itself. That distinction has drawn increasing attention from medical researchers. In humans, uncontrolled cellular reprogramming becomes cancer, growth without regulation or structure. Turritopsis dohrnii appears capable of rebuilding its tissues repeatedly without triggering that collapse into malignancy. Understanding how the jellyfish reorganizes itself while maintaining cellular order could eventually influence oncology, regenerative medicine, and the study of aging itself. And beneath all the laboratory analysis, the animal leaves behind a quieter disturbance. It transformed the life cycle from a straight line into something circular, fluid, and strangely reversible, as though time inside one tiny drifting body had loosened its grip on direction. In the dim layers between sunlight and total darkness, there exists a narrow region where vision becomes uncertain. Light still descends from the surface, but only barely, filtered through hundreds of meters of seawater until the ocean turns blue and indistinct. Shapes above appear only as drifting shadows. Movement matters more than detail. Survival depends on detecting the smallest interruption in the fading glow overhead. Within this suspended world lives a fish called Macropinna microstoma, the barrel-eye fish, moving slowly through depths between roughly 400 and 800 meters. At first encounter, its anatomy appears almost unfinished. The front of the fish is enclosed beneath a transparent dome, a smooth, fluid-filled shield curving over the head like clear glass. Through it, the internal structures of the skull remain visible from the outside. Veins, tissue, and delicate organs can all be seen beneath the surface of the dome, exposed not by injury or decay, but by the animal's normal anatomy. Few vertebrates reveal themselves this openly. Most life protects the head behind bone, muscle, skin, or scales. The barrel-eye turned transparency itself into protection. What appear at first to be its eyes are not eyes at all. Two dark circular spots positioned near the front of the face resemble a familiar pair of forward-looking pupils, but they are only olfactory organs, structures used for detecting chemicals drifting through the water. The real eyes sit deeper inside the skull, hidden beneath the transparent shield. They glow green. Each eye forms a long, tubular structure positioned vertically within the head, shaped almost like a pair of binoculars aimed toward the surface far above. In its resting position, the fish gazes upward constantly. This orientation allows it to detect faint silhouettes crossing the remaining traces of downwelling light. A small crustacean swimming above becomes visible as a moving shadow against the pale blue gradient descending from the world overhead. In the mesopelagic zone, where prey may appear only briefly and disappear just as quickly into darkness, that slight contrast can determine whether the fish feeds at all. The green coloration comes from a yellow pigment embedded within the retina. This pigment filters incoming light and sharpens contrast, helping the fish distinguish between natural sunlight filtering downward and flashes of bioluminescence produced by living organisms nearby. In an environment crowded with cold flashes of biological light, separating one kind of illumination from another becomes essential. The fish does not simply see brightness. It sorts through overlapping sources of light, the way surface animals separate shapes from shadows. For decades, scientists misunderstood how the animal used its vision. Specimens collected from deep water reached the surface badly damaged by decompression. The transparent dome collapsed inward under pressure changes, leaving behind distorted remains that concealed the original structure. Researchers examining preserved specimens saw only the fixed upward-pointing tubes inside the skull and assumed the eyes could not move. Only much later, using submersibles and carefully recorded footage from living individuals, did researchers observe the true mechanism. In 2009, Bruce Robison and Kim Reisenbichler at the Monterey Bay Aquarium Research Institute documented the eyes rotating within the transparent head. While scanning for prey, the fish kept the eyes directed upward, but when moving to feed, the tubular organs pivoted forward, allowing the animal to track targets directly ahead with precise binocular focus. The adaptation solved a problem almost no surface animal has ever faced. In the twilight depths, predators must monitor the dim surface glow above while still navigating forward through near darkness. The barrel eye avoided compromise entirely. It placed rotatable eyes inside a transparent skull, preserving both protection and visibility at once. Over time, the fish quietly unsettled another assumption about anatomy itself. Eyes were once imagined as fixed structures mounted externally onto the body, permanently facing a single direction determined by bone and muscle. Macropinna microstoma dissolved that expectation into something stranger. A visual system suspended within clear tissue, adjustable from within, shaped entirely by the optical geometry of a world where sunlight survives only as memory. In the deeper layers of the ocean, light no longer arrives from the sky. It originates instead from living tissue. A pulse appears briefly in the water, then disappears. Another follows farther away, cold blue drifting through darkness before fading back into black. Across enormous regions of the sea, this is the only illumination that exists, not reflected sunlight or volcanic glow, but light produced directly by organisms themselves through chemistry unfolding inside their bodies. Bioluminescence is the production and emission of light by a living organism through chemical reaction. No electricity powers it, no external flame or stored sunlight fuels it. Inside specialized organs or cells, molecules react together and release energy not as heat, but as visible photons. In the deep ocean, where conserving energy determines survival, the efficiency of the process becomes extraordinary. Almost no warmth escapes. The light appears cool and clean, detached from fire entirely. Scientists estimate that roughly 76% of deep-sea organisms produce some form of bioluminescence. In the largest habitat on Earth, glowing has become ordinary. Entire species communicate, hunt, defend themselves, and disappear into darkness using light generated from within their own biology. On land, luminous animals remain uncommon curiosities. In the deep ocean, darkness itself pushed evolution toward illumination over and over again. The chemistry behind it changes slightly between species, but the central pattern remains remarkably consistent. In many organisms, a molecule called luciferin reacts with oxygen in the presence of an enzyme called luciferase. The reaction releases energy as visible light. Tiny flashes ripple through water where no sunbeam has traveled in millions of years. Most deep-sea bioluminescence appears blue or blue-green because those wavelengths move through seawater more efficiently than any others. Light between roughly 440 and 479 nanometers penetrates farthest before being absorbed. Evolution gradually settled around those colors, the way rivers settle into familiar channels. Red light exists only rarely in the deep, reserved for stranger purposes. Some organisms use bioluminescence not to reveal themselves, but to vanish. The firefly squid, Watacenia scintillans, carries rows of luminous organs across the underside of its body. By matching the faint glow descending from the distant surface far above, the squid erases its silhouette when predators look upward. The animal becomes almost invisible against the diluted light overhead, dissolving its own outline through careful illumination. Other species transformed light into bait. Deep-sea anglerfish drift through darkness, carrying a luminous lure called an esca suspended before their mouths. Curious fish or crustaceans approach the glow instinctively, mistaking it for food or movement, only to disappear into jaws hidden beyond the edge of the light itself. In the vampire squid, Vampiroteuthis infernalis, bioluminescent organs, called photophores, scatter across the body in shifting patterns. The flashes ripple through its webbed form like signals passing silently between stars. Researchers suspect these displays function partly as recognition systems, allowing individuals to identify their own species within the dark water where shape and color become unreliable. Some organisms use light defensively. When attacked, certain shrimp and small fish release clouds of glowing mucus or suspended luminous particles into the surrounding water. The sudden illumination exposes the predator's position, effectively turning the hunter into a visible target for larger animals nearby. Deep-sea biologists sometimes describe this as a burglar alarm strategy, one organism sacrificing concealment long enough to attract something even more dangerous. Then there are the dragonfish. Malacosteus niger, the loose-jawed dragonfish, produces red bioluminescence invisible to most other deep-sea organisms. While surrounding animals evolved eyes sensitive mainly to blue wavelengths, the dragonfish developed the ability to both emit and perceive red light privately. It hunts using what amounts to an invisible searchlight, illuminating prey without the prey realizing illumination has occurred at all. Perhaps the strangest part is how often evolution arrived at this solution independently. Bioluminescence evolved at least 40 separate times across unrelated lineages, appearing again and again in fish, squid, jellyfish, shrimp, worms, and microorganisms. The ocean kept presenting the same problem, and life kept answering with light. Somewhere in the open dark below 200 meters, a small point of light drifts alone through the water. It sways gently, almost delicately, suspended at the end of a thin extension rising from the head of a fish that remains nearly invisible behind it. In the deep ocean, movement toward light is often instinctive. Small animals approach the glow, believing it signals food, safety, or another living presence. By the time they reach it, the mouth waiting behind the light has already opened. More than 200 species of anglerfish exist, but the deep-sea forms belonging to the suborder Ceratioidae became among the most anatomically unusual vertebrates ever discovered. These are the species that evolved bioluminescent lures and drift through the midnight layers of the ocean, where encounters with food may be separated by weeks of emptiness. The female dominates the body plan entirely. In some species, she grows close to a meter in length, her head occupying much of her body, jaws hinged wide enough to engulf prey nearly twice her own size. The stomach stretches with extraordinary flexibility, expanding into a storage chamber capable of holding enormous meals whenever rare opportunities appear. In the deep sea, feeding cannot depend on regularity. Survival favors any anatomy capable of exploiting abundance immediately before scarcity returns. The teeth complete the trap. Long, translucent, and angled inward, they function less like cutting instruments and more like hooks. Once prey crosses the threshold of the mouth, escape becomes mechanically impossible. Every struggle drives the victim deeper inward. Extending above the head floats the esca, the luminous lure itself. It appears almost like a separate organism, a glowing bulb suspended from a modified dorsal spine. The light does not originate from the anglerfish directly. Inside the esca live colonies of bioluminescent bacteria, primarily from the genus Photobacterium. The fish maintains and cultivates these microorganisms within the lure, carrying them as living lanterns whose glow attracts prey through darkness the fish could never illuminate alone. Yet even this predatory anatomy becomes secondary beside the stranger adaptation hidden within the species' reproductive system. The male anglerfish is almost unrecognizable beside the female. In some species, he measures only a few millimeters long, scarcely larger than a mosquito drifting through the water. He possesses no luminous lure, no expandable stomach, no powerful jaws. His body appears simplified nearly to the edge of incompleteness. Large nostrils dominate the head, tuned not for hunting, but for detecting chemical traces released by females across vast distances of open ocean. His entire existence narrows toward a single event. In the deep sea, finding another member of the same species may become one of the rarest encounters an animal ever experiences. Darkness stretches endlessly, populations remain sparse. Individuals drift through enormous volumes of water without guarantee of contact. Evolution answered this isolation with one of the most extreme reproductive strategies known in vertebrate life. When a male finally locates a female, he bites into her body and remains attached. Enzymes released at the contact site dissolve tissue from both animals until their flesh merges together. Blood vessels connect. Circulatory systems fuse into a shared network. Gradually, the male begins to degenerate. His eyes disappear. Internal organs shrink. Much of the brain reduces in complexity. Eventually, little remains except reproductive tissue sustained entirely through nutrients circulating from the female's bloodstream. He becomes part of her body. Some females carry several fused males simultaneously. Up to 8 attached males have been documented connected to a single female serratioid anglerfish, each functioning as a permanent source of sperm whenever reproduction becomes possible. The transformation appears almost impossible from the perspective of ordinary vertebrate biology. Animals are generally defined as separate individuals, maintaining strict boundaries between self and other. Immune systems exist largely to defend those boundaries, attacking foreign tissue before integration can occur. Yet anglerfish evolved mechanisms allowing two separate vertebrates to merge at the level of blood and flesh without rejection. In the stillness of the deep ocean, where encounters become uncertain and isolation stretches across kilometers of darkness, the species abandoned independence itself as a strategy for survival. In the colder layers, between 600 and 900 meters, the ocean passes through a region marine biologists call the oxygen minimum zone. The water there contains so little dissolved oxygen that most active predators cannot survive for long. Respiration becomes difficult, muscles lose efficiency, metabolism slows toward collapse. Yet, drifting through this nearly uninhabitable layer is an animal that seems shaped specifically for it, moving slowly through the dim water with webbed arms spread like dark fabric, suspended in motion. Its name is Vampiroteuthis infernalis, Latin for vampire squid from hell, a title given in 1903 by German marine biologist Carl Chun. The name suggests aggression and blood, but the animal itself behaves with remarkable restraint. It neither hunts actively nor attacks large prey. Even its classification resists expectation. Despite the name, it is not truly a squid, nor is it an octopus. It belongs to its own separate order, Vampiromorphidae, the last surviving member of an ancient lineage that diverged from both squids and octopuses more than 300 million years ago. In appearance, it resembles something assembled from fragments of several different creatures. The body shifts between deep black and dark crimson depending on surrounding light. Webbing stretches between all 8 arms, forming a cloak-like structure that folds and unfurls slowly as it moves. Along the inner surface of this webbing stand rows of fleshy spines called cirri, soft but pointed structures that line the animal's hidden underside like delicate thorns. Its eyes dominate the body. Relative to its size, the vampire squid possesses the largest eyes of any known animal on Earth. In a body measuring roughly 28 centimeters, the eyes alone reach nearly 2.5 centimeters across. They appear deep blue in ordinary conditions but shift toward red beneath certain wavelengths, changing color with the surrounding environment like polished glass catching different light. The enormous eyes help compensate for the faintness of the oxygen minimum zone, but the animal survives there through chemistry as much as vision. Its blood contains hemocyanin, a copper-based oxygen transport molecule that turns blue when oxygenated. While many mollusks use hemocyanin, the vampire squid evolved a version with extraordinarily high oxygen affinity. It can extract usable oxygen from water containing almost none, functioning comfortably in regions where faster predators struggle to breathe at all. By adapting to an environment others abandoned, it gained access to one of the safest habitats in the deep ocean. Even its feeding strategy departs from cephalopod tradition. Squids, octopuses, and cuttlefish are generally active hunters, intelligent predators pursuing fish, crustaceans, and other mobile prey. The vampire squid abandoned that role almost completely. Instead of chasing animals through the water, it extends two long feeding filaments into the surrounding darkness and waits for drifting material to collect upon them. Marine snow settles constantly through the ocean, carrying fragments of dead organisms, fecal particles, mucus, and organic debris downward from surface waters. The vampire squid gathers this falling material carefully, coating it in sticky mucus secreted from its suckers before drawing the collected mass toward its mouth. It survives not through pursuit, but through patience. When threatened, the animal responds with movements unlike those of any true squid. It folds its webbing upward over the body like an umbrella turned inside out, exposing the rows of cirri outward toward the surrounding water while concealing the softer central body within. Instead of releasing ink, which would serve little purpose in darkness, it expels clouds of glowing bioluminescent mucus that drift behind it in shimmering particles, confusing predators while the animal retreats slowly into deeper shadow. The lineage itself appears almost untouched by time. Fossil evidence suggests vampiromorphids have survived for more than 300 million years, with relatively little anatomical change. Ancestors of the vampire squid drifted through ancient oceans long before birds crossed the sky or mammals walked on land. They persisted through mass extinctions, continental shifts, and planetary transformations while remaining hidden inside one of the least hospitable layers of the sea. In the quiet chemistry of the oxygen minimum zone, where faster life struggles simply to breathe, the vampire squid endured by becoming slower, softer, and strangely detached from the violence usually associated with survival. Long before a goblin shark becomes fully visible, its outline appears wrong. The head extends forward into a flattened blade-like structure far longer than expected, while the mouth sits recessed beneath it as though pushed backward into the skull. In dim water, the animal resembles something unfinished, pale and skeletal, drifting through depths where familiar shark anatomy begins to distort into stranger forms. The species is called Mitsukurina austoni, first described scientifically in 1898 from a specimen collected off the coast of Japan, though even now encounters with living individuals remain exceptionally rare. Its skin carries little pigment. Blood vessels show faintly through translucent flesh, giving the body a washed pink or ghostly white coloration, unlike the darker camouflage common among many deep-sea predators. The small eyes appear almost secondary beside the elongated rostrum projecting from the face, a structure that extends well beyond the jaws themselves. At first glance, the snout seems impractical, even cumbersome, but nearly every part of the shark's anatomy exists to solve a problem hidden within darkness. Goblin sharks inhabit depths generally ranging from roughly 270 to 960 meters, though some specimens have been recorded deeper still, beyond 1,300 meters. They occur across the Atlantic, Pacific, and Indian Oceans, yet remain almost invisible to human observation. Fewer than 50 documented encounters with live individuals exist in recorded history, despite adults reaching lengths between 3 and 4 meters. The largest reliably measured specimen exceeded 5 meters, an enormous predator spending most of its existence beyond ordinary visibility. The strangest mechanism reveals itself only when the shark feeds. Most sharks strike by lunging their bodies toward prey, jaws extending modestly forward during the bite. The goblin shark transformed this common mechanism into something far more extreme. Hidden within the skull rests a highly flexible jaw assembly, attached by elastic ligaments and specialized cartilage. When prey enters striking range, the entire jaw launches outward from the face with startling speed, projecting forward almost independently of the body itself. Recorded measurements place the extension velocity at roughly 3 meters per second, making it one of the fastest jaw strikes known relative to body size. The motion appears almost mechanical, as though the shark briefly ejects its own mouth toward the prey before retracting it again. In slow footage, the jaws surge outward, farther than seems anatomically possible, teeth unfolding from beneath the snout in a sudden pale arc. The elongated rostrum serves as more than ornament or hydrodynamic structure. Beneath its surface lies a dense concentration of ampullae of Lorenzini, specialized electroreceptors shared by sharks and rays. These organs detect faint electrical fields produced by muscle contractions and nerve activity in nearby animals. Every swimming fish generates tiny pulses of bioelectricity through movement alone. In the deep ocean, where light becomes unreliable or absent, those signals remain detectable. The goblin shark effectively hunts through electricity. Long before prey becomes visible, the shark senses the weak electrical patterns drifting through surrounding water. A hidden fish twitching inside darkness reveals itself through its own nervous system. Small cephalopods moving nearby announce their presence with every muscular contraction. The shark drifts slowly until the signal sharpens, then the jaws extend forward with explosive precision. Its prey includes teleost fish, crustaceans, and squid, all detected less through sight than through the invisible electrical field surrounding living tissue. Vision remains present, but no longer primary. The predator navigates a sensory world inaccessible to human perception, reading movement through bioelectric disturbance rather than reflected light. The species carries another quiet weight through time itself. Mitsucarina austoni is the last surviving member of the family Mitsucarinidae, a lineage extending back at least 125 million years into the early Cretaceous period. Ancestors of the goblin shark swam through ancient seas, while dinosaurs still dominated terrestrial ecosystems. The body plan endured mass extinctions, shifting oceans, and continental rearrangements with relatively little change. In many ways, the shark appears less like a modern predator than a surviving fragment from another biological era, preserved in deeper water while surface ecosystems transformed repeatedly above it. Its anatomy unsettles familiar assumptions about hunting itself. Predators were once imagined as creatures dependent primarily on sight, pursuing visible targets through direct motion and reaction. The goblin shark reduced vision almost to an accessory sense, replacing it with electrical perception and a strike mechanism capable of crossing the final distance faster than prey can respond. Far beneath the ice-edged waters surrounding Antarctica, there are regions of ocean so cold that movement itself begins to slow. Muscles contract more gradually, chemical reactions lose speed, predators drift rather than chase. In these depths, between roughly 1 and 2 kilometers below the surface, lives an animal so rarely encountered that much of its existence has been reconstructed indirectly from scars on whales, fragments recovered from stomach contents, and brief accidental captures pulled upward from darkness by fishing lines. Its name is Mesonychoteuthis hamiltoni, the colossal squid. It is often confused with the giant squid, Architeuthis ducks, but the distinction matters. Giant squids may reach greater lengths through their elongated feeding tentacles, yet the colossal squid surpasses them in mass. It is the heaviest invertebrate known on Earth. The largest confirmed specimen, hauled accidentally aboard a New Zealand fishing vessel in 2007 and later preserved at the Te Papa Tongarewa Museum in Wellington, measured more than 4 meters and weighed nearly 500 kilograms. Even that individual may not have represented the upper limit of the species. Beaks recovered from sperm whale stomachs suggest much larger animals exist in the Southern Ocean, potentially reaching lengths approaching 14 meters and masses near 750 kilograms, though no intact specimen of that scale has ever been retrieved. The body itself appears built for stillness rather than speed. Thick muscular arms extend outward from a dense central mantle, each lined not with simple suckers but with rotating hooks capable of gripping and tearing simultaneously. Some hooks sit on swiveling bases able to rotate through full circles, locking into prey with mechanical precision. The arms become less like flexible limbs and more like arrays of articulated anchors designed to hold struggling animals in place within dark water. Its eyes are even stranger. The colossal squid possesses the largest eyes ever recorded in any known animal. Fully grown individuals may carry eyes approaching 27 centimeters across, roughly the size of large dinner plates. Embedded within the eye itself rests a specialized light-producing organ called a photophore, creating faint illumination directly beside the visual field. In the darkness of the deep Southern Ocean, such enormous eyes serve a specific purpose. They are not optimized for finding small prey nearby. Instead, they appear adapted to detect distant disturbances. The squid's primary predator is the sperm whale. Far away in black water, a swimming sperm whale disrupts the surrounding ecosystem simply through movement. Smaller organisms react. Bioluminescent creatures flash and scatter as the whale approaches. An eye large enough to detect those faint changes from more than 100 meters away may provide the colossal squid with enough warning to escape before direct contact occurs. The immense eyes evolved not primarily for hunting, but for survival against something even larger. Most direct evidence of these encounters comes from the whales themselves. Sperm whales frequently carry circular scars across their skin, marks left behind by the squid's rotating hooks during violent struggles far below the surface. Entire battles unfold unseen in darkness beneath Antarctic waters, their existence revealed only through scars drifting slowly across the skin of surfacing whales. What unsettled biologists further was not simply the squid's size, but how little energy it appears to require. A study published in 2020 suggested the colossal squid possesses an extraordinarily slow metabolic rate. Despite its immense mass, it may survive on only a few dozen grams of food each day. In the food-poor depths of the Southern Ocean, this efficiency becomes essential. Large size normally demands abundant caloric intake. The colossal squid solved the problem differently, slowing itself almost to the edge of stillness. Its entire body remains specialized for cold. The surrounding water near its preferred depth hovers close to freezing, generally between 0 and 2 degrees Celsius. Muscle tissue evolved specifically for function under those conditions. In warmer surface temperatures, the squid's movements would likely become impaired, its physiology drifting outside the narrow thermal range where it can operate effectively. Everything about the animal contradicts familiar assumptions about scale. On land, large predators require constant feeding, rapid circulation, and immense energetic throughput. The colossal squid reached gigantic proportions not by accelerating its biology, but by slowing it down. In the deep Antarctic Ocean, efficiency replaced abundance, and stillness became powerful enough to support one of the largest bodies evolution ever assembled without a backbone. Deep beneath the ocean floor, seawater slips slowly through fractures in the Earth's crust and descends toward buried magma chambers. As it sinks, the water becomes trapped within superheated rock, absorbing dissolved metals, gases, and minerals before rising again through cracks in the seafloor. When it finally erupts back into the ocean, it emerges as a hydrothermal vent, a rupture in the planet where seawater and volcanic chemistry meet in continuous exchange. The temperatures surrounding these vents reach extremes difficult to imagine beneath the ocean. Some vent fluids emerge at more than 400 degrees Celsius, hot enough to glow with shimmering distortion as they enter the freezing deep water around them. Yet the water does not boil. Under ordinary conditions at the surface, liquid water transforms rapidly into vapor at 100 degrees Celsius. In the abyss, pressure changes the rules entirely. The immense weight of the ocean pressing downward raises water's boiling point so dramatically that near molten temperatures remain liquid within the deep sea itself. Different vent systems produce different forms of eruption. Black smokers release dark, mineral-rich plumes saturated with sulfide particles and heated to the highest temperatures yet measured in the deep ocean. Around the vent openings, dissolved minerals cool and solidify into towering chimney structures that rise from the seafloor like blackened stone pillars. White smokers erupt at lower temperatures and carry different chemical mixtures rich in compounds containing barium, calcium, and silicon, producing pale mineral clouds that drift more softly through the surrounding water. Everything about the vent fluid appears hostile to ordinary surface biology. Hydrogen sulfide, methane, dissolved metals, carbon dioxide, manganese, zinc, copper, and iron concentrate within the erupting water at levels toxic to most familiar organisms. Yet around these poisonous plumes, life gathers with extraordinary density. The foundation belongs not to plants, but to microorganisms. Chemosynthetic bacteria and archaea coat the vent surfaces in thick microbial films, extracting energy directly from the surrounding chemicals. They transform dissolved compounds into organic material the way forests transform sunlight into living tissue. Entire ecosystems rise upon this invisible chemistry. The bacteria become food, habitat, and biological infrastructure all at once. Towering above them grow giant tube worms, Riftia pachyptilla, their pale tubes clustered tightly against the vent chimneys. Some individuals exceed 2 meters in length, surviving entirely through the bacterial symbionts living inside their bodies. Nearby, Pompeii worms construct thin tubes directly against the walls of black smoker structures, where temperatures may approach 80 degrees Celsius, making them among the most heat-tolerant complex animals ever documented. Their backs carry dense bacterial coatings that may help insulate the worms from thermal extremes while also contributing nutritionally. Elsewhere, among the vents crawl yeti crabs, Kiwa hirsuta, discovered only in 2005 in the South Pacific. Long blonde setae cover their claws and limbs like pale fur drifting in the current. Upon those hair-like structures grow colonies of chemosynthetic bacteria that the crabs cultivate and harvest continuously, farming microorganisms directly upon their own bodies. Even fish adapted to vent ecosystems tolerate conditions that would destroy most vertebrate proteins. Zooarcid vent fish graze along bacterial mats near temperatures capable of denaturing ordinary biological molecules, surviving within gradients of heat and chemistry once assumed impossible for vertebrate tissue. Yet these ecosystems remain temporary on geological timescales. A hydrothermal vent field may persist for decades or centuries before volcanic activity shifts elsewhere beneath the crust. Once the heat source fades, the vent weakens, chemical production collapses, and the surrounding biological community dies with it. Entire ecosystems disappear within only a few years after vent activity ceases. New vent systems eventually emerge elsewhere along the ocean floor, often separated by hundreds of kilometers of barren seafloor. Somehow larvae from vent organisms locate these isolated habitats through darkness and open ocean distance. Scientists still do not fully understand how such dispersal occurs with enough reliability to sustain vent populations across disconnected volcanic systems. The vents revealed something larger than unusual creatures alone. They exposed ecosystems built entirely from conditions once considered fatal, where poison became energy, volcanic instability became habitat, and life arranged itself around the chemistry of the planet's interior rather than the distant light of the sun above. Along the fractured volcanic ridges of the East Pacific Rise, black smoker chimneys rise from the seafloor like burning towers frozen beneath the ocean. Mineral-rich water erupts continuously from their surfaces, carrying temperatures hot enough to soften metal and strip oxygen from surrounding water. Pressed directly against these chimneys, where the boundary between liquid water and geological heat narrows to only a thin margin, lives a small gray animal named Alvinella pompejana, the Pompeii worm. It received its name from the buried Roman city, destroyed by volcanic eruption nearly 2,000 years ago. An echo carried from land into the deep Pacific in 1980 when scientists first described the species. The comparison was not poetic exaggeration. Few animals on Earth exist so close to environmental conditions that resemble catastrophe. The Pompeii worm builds fragile paper-thin tubes directly onto the outer walls of active black smoker chimneys. The tubes cling to surfaces already coated in mineral deposits and bacterial films, placing the worms only centimeters from vent fluids heated far beyond the tolerance limits of ordinary biology. Inside these narrow shelters, the worm positions its body across one of the most extreme temperature gradients known in any multicellular organism. Its tail remains deep within water, approaching 80 degrees Celsius, while its head extends outward into surrounding seawater closer to 22 degrees Celsius. Across the length of a single body exists a thermal difference of nearly 60 degrees, a range greater than the environmental variation experienced across entire ecosystems at the surface. One portion of the animal lives in conditions approaching near boiling heat, while another occupies temperatures cold enough to preserve the stability of its tissues. This arrangement creates an enormous biological problem. Proteins, the molecular machinery responsible for almost every function inside living cells, begin to unfold and fail when exposed to excessive heat. In most animals, temperatures above 40 or 50 degrees Celsius permanently damage enzymes, disrupt cellular membranes, and collapse metabolic systems. Yet, the Pompeii worm continues functioning where such limits should already have been crossed. Inside, its cells are specialized heat shock proteins and thermostable enzymes capable of maintaining their structure under temperatures that would destroy equivalent proteins in nearly every other known animal. These molecules stabilize cellular processes even while heat pulses continuously through portions of the worm's body. Rather than collapsing under thermal stress, its biology appears calibrated to operate within it. The worm carries another unusual feature across its back. Dense colonies of filamentous bacteria form pale, layered mats over its skin, resembling soft fleece drifting gently in vent currents. For years, scientists debated whether these bacterial coatings represented accidental contamination from the surrounding vent environment or an intentional partnership between animal and microbe. Gradually, evidence began suggesting the relationship was far more intimate. The bacteria likely provide partial thermal insulation, forming a living barrier between the worm and surrounding chemical extremes. They may also help neutralize toxic sulfide compounds dissolved within vent water, reducing the chemical stress placed upon the animal itself. Some researchers suspect the bacteria contribute nutritionally as well, though the precise balance of the relationship remains only partly understood. The enzymes functioning inside the Pompeii worm have drawn increasing attention far beyond marine biology. Heat-stable enzymes already play a central role in polymerase chain reaction technology, the DNA amplification process used in modern medical diagnostics, forensic analysis, and molecular genetics. Organisms adapted to hydrothermal vents provide entirely new variations of these biochemical tools, expanding the temperature ranges and stability limits available to biotechnology. Yet studying the worm directly remains extraordinarily difficult. Every attempt to retrieve living specimens from the vents has ended in failure. The combined shock of pressure loss and temperature change kills the animals before controlled laboratory measurements become possible. Most scientific understanding comes instead from instruments lowered directly into the vent environment itself, recording data within the darkness where the worms naturally survive. The Pompeii worm altered one of biology's oldest assumptions. Complex animal life was once believed incapable of functioning near the thermal boundaries of liquid water. Pressed against volcanic chimneys beneath the Pacific, Alvanella pompejana quietly crossed that boundary and remained there. Nothing in the deep ocean remains untouched by pressure. It presses against skin, bone, membranes, blood, and molecules themselves with a force so constant that life there has never experienced the absence of it. Every descent through seawater adds more weight from the ocean above. For roughly every 10 meters of depth, pressure increases by another atmosphere. At 10,000 meters beneath the surface, the pressure rises to nearly 1,000 atmospheres, approaching 100 megapascals, enough force to crush most human engineering into collapse. Simple objects reveal the effect immediately. A styrofoam cup lowered into the abyss returns compressed into something barely recognizable, its trapped air squeezed inward until the cup shrinks to the size of a small thimble. Hollow spaces disappear first. Air pockets vanish. Any structure dependent on internal emptiness collapses under the surrounding weight. Biology faces the same problem at every scale. Living cells are enclosed by membranes, built from lipid bilayers, delicate structures whose flexibility allows nutrients, signals, and proteins to move properly through the cell. Under extreme pressure, these membranes stiffen, Their fluidity decreases until they begin behaving almost like frozen surfaces, disrupting communication between cells and interfering with the proteins embedded within them. In ordinary surface organisms, such pressure would destabilize metabolism itself. Cellular systems would fail long before reaching the deepest trenches. Deep-sea organisms solve this problem not through resistance, but through chemical redesign. Their membranes contain far higher concentrations of unsaturated fatty acids, molecules whose bent structures prevent the membrane from packing too tightly together. Even under crushing pressure and near freezing temperatures, these membranes remain flexible enough to function. The chemistry of the deep-sea cell is subtly different from that of surface life, adjusted molecule by molecule for conditions that surface biology was never built to survive. Pressure creates another danger beyond membrane stability. Proteins, enzymes, and strands of DNA all depend upon precise three-dimensional shapes to function correctly. High pressure distorts those shapes, interfering with folding patterns essential for life. To counter this, deep-sea organisms produce special protective molecules known as piezolites, compounds that stabilize cellular structures against compression. Among the most important is trimethylamine oxide, commonly called TMAO. In Hydrothermal living below 6,000 meters, TMAO concentrations rise to extraordinary levels. The molecule acts almost like a microscopic brace, helping proteins maintain their functional shapes despite the immense pressure surrounding them. The Hadal snailfish, Pseudoliparus swiri, carries some of the highest concentrations ever measured. Collected from depths exceeding 8,000 meters, its tissues contain so much TMAO that the flesh reportedly carries a strong ammonia-like taste. Measurements show a remarkably consistent pattern across deep-sea species. The deeper the fish lives, the more TMAO accumulates within its cells, as though pressure itself directly determines the chemistry of survival. Yet even this adaptation appears to approach a limit. Biochemical models suggest that beyond a certain concentration, TMAO begins disrupting cellular processes rather than protecting them. The molecule that stabilizes proteins eventually becomes chemically burdensome in excess. This may explain one of the quiet boundaries of vertebrate life in the ocean. Although trenches descend beyond 10,000 meters, no fish have ever been confirmed below roughly 8,400 meters. Beneath that depth, pressure may push vertebrate biochemistry beyond what even piezolites can compensate for. The bodies of deep-sea fish reflect the same pressures externally. Many species possess minimal bone structure, with partially cartilaginous skeletons and gelatinous flesh that reduces internal pressure differences between tissues and surrounding water. Rigid structures become liabilities under such conditions. Softness becomes protection. The deeper scientists looked, the more pressure revealed itself not as a simple environmental factor, but as a force capable of reshaping life at the molecular level. Proteins, membranes, enzymes, and skeletal structures all required modification before survival became possible. The deep sea did not merely challenge biology, it forced biology to rebuild itself from the inside outward. At the bottom of the Mariana Trench, where the seafloor descends beyond nearly every known limit for vertebrate life, a small translucent fish moves slowly through the sediment clouds of the Hadal zone. It does not resemble the powerful predators associated with the deep ocean. There are no enormous teeth, no armored scales, no dramatic bioluminescent lures. The deepest known fish on Earth appears delicate, almost unfinished, as though ordinary biology had been softened by the pressure surrounding it. Its name is Pseudoliparus swiri, the Mariana snailfish. First formally described in 2017, the species holds the confirmed record for the deepest fish ever physically collected, recovered from approximately 8,336 meters below the Pacific Ocean surface. In 2023, researchers associated with the University of Western Australia recorded additional footage of a snailfish at roughly the same depth, possibly representing another record individual. Even at the edge of vertebrate survivability, the trench still carried movement. The fish itself remains surprisingly small. Most individuals measure only between 20 and 28 centimeters long. Their bodies are pale and nearly transparent, with gelatinous flesh and minimal skeletal rigidity. Internal organs faintly show through the thin skin beneath submersible lights, giving the animal the appearance of something only partially separated from the surrounding water. The body narrows into a tadpole-like shape, with soft fins that drift rather than cut through the water. At such depths, rigidity becomes dangerous. The swim bladder found in most fish, a gas-filled organ used for buoyancy control, would implode almost instantly under hadal pressure. The snailfish has abandoned the structure entirely. Instead, buoyancy emerges from the chemistry and density of its tissues themselves. Its flesh remains soft, watery, and low in density, reducing the pressure differences that rigid compartments would create inside the body. In the trench, softness becomes structural stability. Its survival also depends upon the molecular adaptations carried within its cells. The trimethylamine oxide compounds discussed earlier accumulate in extraordinarily high concentrations inside hadal snailfish tissues, stabilizing proteins against the immense pressure pressing inward from all sides. Without these compounds, cellular machinery would distort and fail. The fish exists near the upper limit of what vertebrate biochemistry appears capable of tolerating. Around the trench floor, amphipods swarm in surprising abundance, feeding upon the marine snow drifting downward through the water column. Ocean trenches act like enormous collection funnels, concentrating organic debris from vast regions above. The snailfish feeds primarily upon these small crustaceans, moving through the sediment with quiet efficiency. In an environment where food remains scarce and unpredictable, the trench itself gathers enough falling material to support entire hadal communities. Vision becomes increasingly unimportant at such depths. Sunlight disappeared thousands of meters above, leaving only darkness interrupted occasionally by distant bioluminescent pulses. The snailfish instead relies heavily upon enlarged lateral line canals running along its body, pressure-sensitive organs capable of detecting minute vibrations and water movement. Through these sensory structures, the fish perceives nearby motion in a world where ordinary sight has little value. Even reproduction occurs within the trench itself. Unlike many marine animals whose larvae drift upward into shallower waters, hadal snailfish complete their entire life cycle under crushing pressure. Their eggs are comparatively large and rich in yolk reserves, suggesting an evolutionary strategy centered on producing fewer offspring with greater developmental support. Survival in the trenches leaves little room for waste, yet beneath the quiet success of the snailfish lies a remarkable biological boundary. No confirmed fish has ever been found significantly deeper than approximately 8,400 meters. Despite ocean trenches extending more than 2,000 meters below that threshold. The deepest portions of the Mariana Trench may remain permanently inaccessible to vertebrate life, not because deeper water cannot physically exist, but because the chemistry required to stabilize proteins eventually becomes impossible to maintain. The snailfish drifts precisely along that boundary, occupying the final narrow layer where fish biology still functions before pressure closes the door completely. Far below the reach of storms and tides, An octopus drifts through the water with the slow rhythm of something suspended inside a dream. Its fins rise and fall gently from either side of its mantle, moving not with urgency, but with quiet repetition, as though the animal has learned that speed carries little value in the stillness of the abyss. In the glow of submersible lights, the silhouette resembles a small floating cloak with wide ears extending outward into darkness. Scientists placed it within the genus Grimpoteuthis, though it became known more widely as the Dumbo octopus. There are 13 recognized species within the group, scattered through deep ocean habitats across the planet. The nickname emerged from the broad ear, like fins attached to the mantle, a resemblance to the animated elephant from surface culture carried unexpectedly into one of the least human places on Earth. The Dumbo octopus lives deeper than any other known octopus. In 2020, researcher Alistair Graham and colleagues documented a confirmed sighting at nearly 6,957 meters below the ocean surface, placing the animal within the hadal depths, where only a small number of complex creatures continue to survive. Even among deep-sea cephalopods, it occupies one of the most inaccessible layers of the planet. Most individuals remain modest in size, measuring roughly 20 to 30 centimeters across, though rare specimens have reached lengths approaching nearly 2 meters and weights exceeding 5 kilograms. Their bodies appear soft and rounded, lacking the muscular density associated with shallow water octopuses that hunt aggressively among reefs and rocky coastlines. Movement in the Dumbo octopus reflects the deep sea itself. Rather than propelling itself with rapid jet bursts through the siphon, as most octopuses do, it drifts through the water column using the slow undulation of its fins. Its arms trail gently behind or adjust subtly for balance and maneuverability. The entire body seems adapted toward conserving energy rather than explosive motion. Much of traditional octopus anatomy has been simplified. The Dumbo octopus possesses no ink sac at all. In shallow water species, ink functions as a visual defense, a cloud of darkness released to confuse predators in illuminated environments. In the perpetual darkness of the deep ocean, such a defense would carry little purpose. Evolution simply removed it. Its feeding behavior has simplified as well. Most octopuses use powerful beaks and rasping radulas to tear prey apart before consumption. The Dumbo octopus instead swallows prey whole. Small crustaceans, worms, and other seafloor animals disappear directly into the mouth with minimal processing. The radula itself has become reduced, reflecting prey that no longer requires dismemberment before digestion. One of the most revealing discoveries about the animal emerged from the study of a developing egg recovered from the deep ocean. Inside was not a drifting larval form, but a miniature, fully developed Dumbo octopus already resembling the adult animal. Unlike most cephalopods, which hatch into vulnerable larvae that spend part of their lives drifting through shallower waters, the Dumbo octopus bypasses the larval stage entirely. This change carries enormous importance in the deep sea. Larvae drifting upward or sideways through ocean currents risk entering environments where pressure, temperature, or predators become immediately lethal. By hatching as complete miniature adults already adapted to abyssal conditions, the Dumbo octopus eliminates one of the most dangerous phases of marine life. The surrounding environment remains remarkably stable compared to the surface world. Temperatures across much of the deep ocean fluctuate only between roughly 2 and 10 degrees Celsius, forming a narrow thermal band that changes little across vast distances and geological timescales. Within that stability, The Dumbo octopus evolved towards simplicity rather than complexity of behavior. It bends another assumption carried from surface biology. Cephalopods are often treated as symbols of active intelligence, rapid predation, and constant interaction with dynamic surroundings. The Dumbo octopus survives through almost the opposite strategy. It drifts, conserves, simplifies, and reproduces quietly in darkness, so complete that motion itself becomes slow enough to resemble stillness. Not all deep-sea ecosystems erupt from violence. Some emerge so slowly that the surrounding seafloor appears almost motionless, with no towering chimneys, no explosive plumes, no visible release of heat into the water. Beneath layers of sediment, hydrocarbons migrate upward through buried geological formations, rising patiently through cracks and porous rock until they finally seep into the ocean itself. These places are known as cold seeps. The fluids escaping from the sediment carry methane, hydrogen sulfide, brine, and dissolved hydrocarbons generated deep within the earth. Unlike hydrothermal vents, the temperature of these fluids remains close to the surrounding seawater. There is no dramatic thermal contrast, only chemistry slowly entering the deep ocean from below. Scientists first recognized cold seeps in 1984 within the Gulf of Mexico, when oceanographer Charles Paul and colleagues aboard the research vessel Sea Probe encountered unusual biological communities nearly 3,200 meters beneath the surface. The seafloor there held organisms clustered around areas where hydrocarbons leaked continuously from below, revealing another ecosystem built not from sunlight, but from chemical energy. The geological origins of cold seeps are tied to the same deep processes that create petroleum reservoirs beneath continents and ocean basins. Organic material buried within sediment over millions of years transforms into hydrocarbons under pressure and heat. In certain locations, these materials migrate upward until pathways through the sediment allow them to escape into the water column. Wherever that chemical leakage reaches the seafloor, life gathers around it. The biological structure of cold seep communities resembles hydrothermal vent ecosystems in broad outline. Chemosynthetic bacteria form the foundation, converting dissolved chemicals into organic energy that supports larger organisms above them. Yet, the atmosphere of a seep differs profoundly from the volcanic instability of vents. Cold seeps are quieter, slower, and vastly longer-lived. Among the most remarkable residents are the seep tube worms, Lamellibranchia lumesi, anchored in dense clusters around seep sites. These animals grow upward in pale branching formations, sometimes called tube worm bushes. Individual worms are estimated to survive for more than 200 years, making them among the longest-lived animals on Earth. Their growth is slow and deliberate, accumulating over centuries in environments where chemical conditions remain stable across immense stretches of time. Elsewhere on the Gulf of Mexico seafloor, another strange adaptation appears directly upon methane hydrate itself. Methane hydrate is a frozen ice-like material formed when methane gas becomes trapped within crystalline water structures under cold temperature and high pressure. Living directly upon this hydrocarbon solid are methane ice worms, Hesiocaca methanicola. The worms burrow into the methane hydrate and appear to graze upon bacterial communities coating its surface. Few animals anywhere on Earth live directly upon a substance so chemically alien to ordinary biology. Some cold seep systems produce even stranger landscapes. Hypersaline brine emerging from beneath the sediment can become between 3 and 8 times saltier than ordinary seawater. Because the brine is denser than the surrounding ocean, it pools directly upon the seafloor in formations that resemble underwater lakes. Their edges form visible shorelines. Submersibles, disturbing the surface, create ripples and waves moving across the dense liquid boundary. The effect upon ordinary marine animals can be immediate. Creatures entering the brine often become stunned or die from osmotic shock, as the extreme salinity disrupts cellular balance. Yet around the edges of these submerged lakes, microbial life flourishes. Specialized organisms inhabit the narrow transition zones between normal seawater and concentrated brine, occupying chemical boundaries that would kill most other animals within moments. What distinguishes cold seep ecosystems most profoundly is their longevity. Hydrothermal vents depend upon volcanic heat sources that eventually shift or collapse, ending the ecosystems surrounding them. Cold seeps, by contrast, can remain active for tens of thousands of years. Their chemistry flows from geological reservoirs buried deep within the earth, sustaining stable communities across timescales longer than many surface ecosystems survive. In the deep ocean, life does not always arrive with eruption or fire. Sometimes it gathers slowly around invisible chemical leaks, building entire ecosystems through patience alone. Far below the abyssal plains, where even the deep ocean begins to thin into something more isolated and severe, the seafloor breaks apart into trenches that descend beyond 6,000 meters. This region is known as the Hadal Zone, named after Hades, the underworld of Greek mythology. Across the planet, there are 37 known ocean trenches, most carved into the Pacific Ocean along tectonic boundaries, where one section of Earth's crust bends and sinks beneath another. These trenches are not narrow cracks, but immense geological structures, long valleys folded downward into darkness, deeper than any mountain rises into the sky. The most famous of them lies in the western Pacific Ocean, east of the Mariana Islands. Within this trench sits Challenger Deep, the deepest known point on Earth's surface, descending approximately 10,935 meters below sea level. The scale resists ordinary comparison. If Mount Everest were lowered intact into the Mariana Trench, more than 2,000 meters of water would still cover its summit. The deepest point of the ocean is not merely a little deeper than the highest mountain is tall. It exceeds it by a distance large enough to contain entire additional landscapes above it. At these depths, the ocean becomes almost entirely separated from surface experience. Pressure rises beyond 1,000 atmospheres. Temperatures hover near freezing. Sunlight has never existed there. Food drifts downward, only in fragments from worlds far above, yet life persists. The Hadal Zone supports bacteria and archaea living within sediment and rock, alongside fungi, shelled foraminifera, polychaete worms, sea cucumbers, amphipods, and the pale snailfish already encountered in the trench depths. Many of these organisms survive through extraordinary biochemical adaptations that protect membranes, proteins, and cellular structures from pressures capable of crushing surface biology into collapse. Among the most abundant animals are hadal amphipods, such as Hirondelia gigas. These shrimp-like crustaceans gather in astonishing numbers around baited traps lowered into trenches. Expeditions to the Mariana Trench have recorded thousands arriving within only a few hours, their bodies swarming through darkness around any available source of organic matter. For decades, scientists assumed biomass would decline steadily with increasing depth, yet trenches repeatedly contradicted that expectation. Part of the explanation lies in the shape of the trenches themselves. Their steep-sided geometry acts like a funnel across immense areas of surrounding ocean floor. Marine snow, dead plankton, Decaying tissue, fecal particles, and drifting organic debris settle downward through the water column and become concentrated within the trench bottom. Rather than starving wastelands, some hadal regions accumulate more food than the surrounding abyssal plains, creating narrow pockets where surprisingly dense ecosystems can survive. But the same downward funnel that gathers nourishment also gathers contamination from the modern surface world. In 2017, researcher Alan Jameson and colleagues discovered extremely high concentrations of polychlorinated biphenyls and polybrominated diphenyl ethers inside amphipods collected from the Mariana and Kermadec trenches. These industrial chemicals, banned decades ago in many countries, had descended through the ocean and accumulated within some of the deepest animals on Earth. Concentrations measured in the trench amphipods exceeded levels documented in heavily polluted rivers near industrial centers in China. Even here, nearly 11 kilometers beneath the surface, the chemistry of human industry had arrived first. Human beings themselves have reached Challenger Deep only a handful of times. In 2012, filmmaker and explorer James Cameron descended alone in the deep-sea Challenger submersible, spending approximately 3 hours upon the trench floor. He described a flat expanse of pale sediment stretching outward beneath the submersible lights, seemingly empty and silent. Yet biological samples, taken from the same depths, revealed an active microscopic world embedded within the mud itself, invisible from a viewport yet fully alive. In the hadal zone, life persists beyond the limits once imagined possible, gathering within trenches shaped by tectonic violence and fed by the slow, falling remains of the brighter ocean far above. Long before human beings named the oceans, before mammals walked on land, before flowering plants spread across continents, a small, armored, crustacean body plan had already taken shape beneath ancient seas. That design still exists, almost unchanged today in Bathynomus giganteus, the giant isopod, the largest known member of the isopod order. Its distant relatives include the small woodlice that move quietly beneath stones and fallen leaves in gardens around the world. Yet, in the deep ocean, the same blueprint expanded into something far larger and stranger, as though an ordinary creature had been rebuilt at the scale of another age. A fully grown giant isopod can reach nearly 50 centimeters in length and weigh as much as 1.7 kilograms, large enough to resemble a pale marine armadillo resting against the sea floor. It inhabits depths between roughly 170 and 2,140 meters across parts of the Atlantic, Pacific, and Indian Oceans, moving slowly over muddy or clay-rich sediment where sunlight vanished long above. Its body appears assembled from overlapping plates of armor, each segment fitting into the next with ancient mechanical precision. 7 pairs of legs carry it across the seafloor. 2 pairs of antennae sweep through the water ahead, sampling faint chemical traces invisible to every human sense. Compound eyes, containing approximately 4,000 facets, stare outward from the sides of the head, adapted not for clarity, but for gathering the smallest fragments of dim light that sometimes descend from above. When disturbed, the animal can curl inward into a nearly perfect defensive sphere, its armored plates sealing together with the same strategy used by tiny pillbugs beneath surface stones. The giant isopod survives primarily as a scavenger. In the deep sea, death becomes one of the most important food sources. A dead fish, squid, or whale descending from upper waters can feed entire communities for weeks. Giant isopods detect these events chemically following dissolved signals through darkness until they arrive before competing scavengers. Their jaws are capable of tearing soft tissue, and they have also been documented feeding on slow-moving live animals such as sea cucumbers when opportunity permits. Yet meals in the deep ocean arrive unpredictably. Days may pass without food. Months may pass. Entire seasons may drift by in darkness, with nothing substantial appearing from above. The giant isopod survives this instability through a metabolism so slow that its existence seems almost suspended. In captivity at Toba Aquarium in Japan, one giant isopod survived without eating for 5 years and 43 days before eventually dying. No other animal observed under controlled conditions has maintained such a prolonged fast. Cold temperatures contribute to this remarkable endurance. Near freezing water slows chemical reactions throughout the body, reducing energy demands to extraordinary lows. Fat reserves are conserved carefully, movement remains minimal, and digestion itself can stretch across immense spans of time. In the deep ocean, survival often favors patience over speed. The broader shape of the animal carries another quiet revelation. Fossils from the Jurassic period reveal isopods almost indistinguishable from living giant isopods today. Across approximately 160 million years, through asteroid impacts, continental rearrangements, and mass extinctions, the essential design remained stable. Evolution did not continuously reshape it into something more elaborate. The existing structure already functioned effectively within an environment that changed very little. Scientists still debate why deep-sea isopods became so much larger than their shallow water relatives. One explanation points toward cold water slowing metabolism and extending growth periods over many years. Another suggests that oxygen dissolves more efficiently in cold water, supporting larger body sizes. Reduced predation pressure may also allow animals to survive long enough to reach dimensions impossible in crowded surface ecosystems. In the deep sea, where conditions remain stable across geological ages, evolution sometimes stops searching for new solutions. A form that works simply continues, patient and unchanged, while entire worlds above it rise and disappear. Through the lights of a descending submersible, the deep ocean often appears, filled with drifting white particles, endless flecks moving downward through darkness like slow winter weather beneath the sea. Oceanographers named this phenomenon marine snow because of that resemblance, though each falling particle is not frozen water, but the remains of living systems from far above. The deep ocean survives on this constant silent precipitation, a continuous downward movement of organic matter linking the bright surface world to the abyss below. Marine snow is composed of dead phytoplankton, zooplankton, fecal pellets, discarded exoskeletons, bacterial colonies, fragments of jellyfish and fish tissue, clay minerals, atmospheric dust, and strands of mucus released by microscopic organisms. Much of it becomes bound together by sticky transparent substances known as transparent exopolymer particles, delicate mucus threads that cause tiny fragments to clump into larger drifting masses. Illuminated in darkness, the particles resemble ash suspended in water, though each fragment carries stored energy and carbon from sunlight captured near the ocean surface. The descent is slow. Individual particles sink at rates between roughly 100 and 150 meters per day. Organic material created in surface waters may require weeks to reach the deep ocean floor. During that journey, the material changes continuously. Bacteria colonize it. Midwater organisms intercept and consume portions of it. Nutrients dissolve outward into surrounding water. By the time the remains reach abyssal sediments, Only a small fraction of the original organic carbon survives intact. Yet that fraction sustains entire ecosystems. This movement of carbon from surface waters into the deep ocean forms what scientists call the biological pump. Near the surface, phytoplankton use sunlight through photosynthesis to capture carbon dioxide from the atmosphere and convert it into living tissue. Small animals consume the phytoplankton. Larger animals consume them in turn. When organisms die, excrete waste, or shed biological material, carbon begins sinking downward through the ocean interior. Some eventually becomes buried within deep-sea sediments, isolated from the atmosphere for centuries or even millennia. Every year, the biological pump transfers an estimated 5 to 12 billion tons of carbon from the upper ocean into the deep sea. Without this process, atmospheric carbon dioxide concentrations would be dramatically higher than they are today. The deep ocean does not merely store water and strange life. It regulates planetary chemistry on a scale large enough to shape climate itself. Even so, very little marine snow reaches the abyss untouched. Deposit feeders sift sediment continuously for organic fragments. Scavengers roam across the seafloor, searching for concentrated falls of nutrition. Bacteria consume nearly everything remaining. In the deep ocean, almost no energy can be wasted. Occasionally, the descent becomes far larger than drifting particles. When a whale dies and sinks into the abyss, its body creates what marine biologists call a whale fall, a concentrated delivery of organic matter capable of sustaining entire ecological communities for decades. The carcass first attracts mobile scavengers such as sharks and amphipods that strip soft tissue from the skeleton. Later, enrichment opportunists colonize the surrounding sediment, feeding on material embedded in the sea floor. Eventually, the bones themselves become ecosystems. Among the strangest inhabitants of this final stage are Osidax worms, discovered in 2002. Their name means bone eater. These small worms possess no mouth and no digestive system. Instead, they extend root-like structures into whale bones, secreting acid that dissolves the mineral matrix. Symbiotic bacteria living within the worms process the released lipids and nutrients, feeding the animal indirectly through chemistry, much like the tube worms surrounding hydrothermal vents. The biological pump binds the surface ocean and the abyss into a single connected system. Changes near the surface, warming waters, acidification, collapsing fisheries, altered plankton populations, ripple downward through every stage of the descent. If the pump weakens, less carbon reaches the deep sea, and more remains in the atmosphere above. Far beneath the waves, in darkness untouched by sunlight, the ocean continues performing one of the most important climate processes on Earth through nothing more dramatic than the slow, endless fall of marine snow. In the middle depths of the ocean, where the last traces of surface light dissolve into permanent twilight, vision becomes a contest shaped almost entirely by bioluminescence. Animals flash signals, mimic drifting particles, erase their silhouettes with counter-illumination, or lure prey through carefully controlled pulses of blue light. Across these waters, nearly every species shares the same visual language because seawater carries blue-green wavelengths farther than any other color. Evolution built an enormous hidden ecosystem around that single spectrum. Then one predator stepped outside the system entirely. The deep-sea dragonfish family, Stomiidae, contains approximately 270 known species, distributed between depths of roughly 200 and 2,000 meters. Many possess elongated bodies, fang-like teeth, and rows of glowing photophores extending across their flanks. Yet among them, Malacosteus niger The loose-jaw dragonfish developed one of the strangest sensory adaptations in the ocean. Its body is relatively small, usually reaching only 25 to 30 centimeters in length, but the head appears almost skeletal. The lower jaw hangs beneath the skull without a floor or membrane joining the bones together. Instead of forming a sealed structure like the jaws of most fish, it remains partially open, an exposed framework capable of swinging downward to extraordinary angles with minimal resistance from surrounding water. In the dense, cold ocean, even slight reductions in drag matter. The loose jaw can open its mouth rapidly without pushing a large wall of water ahead of it. The fish occupies the mesopelagic and bathopelagic zones, regions where nearly all visual communication relies on blue-green bioluminescence. Most deep-sea animals produce light in wavelengths between approximately 440 and 479 nanometers, because these colors travel efficiently through seawater. Eyes throughout the deep ocean evolved accordingly. Predators, prey, scavengers, and drifting organisms all became tuned to the same narrow band of visible light. Malacosteus niger evolved beyond it. The loose-jaw dragonfish produces far-red and near-infrared bioluminescence at wavelengths approaching 700 nanometers and above, colors almost no other deep-sea animal can perceive. At such depths, red light normally does not exist. Surface sunlight loses its red wavelengths within the upper ocean long before reaching the twilight zone. For millions of years, most deep-sea creatures simply stopped evolving sensitivity to red entirely because there was no environmental reason to retain it. As a result, the dragonfish effectively carries an invisible searchlight. Small crustaceans, such as copepods, drift through darkness, scanning constantly for flashes of approaching blue-green bioluminescence that might signal danger. Yet the red illumination emitted by Malacosteus passes unnoticed through their visual systems. The predator can illuminate prey directly while remaining functionally unseen, watching movements hidden from every surrounding animal. To perceive its own unusual light, the dragonfish evolved a unique retinal adaptation. Its eyes contain a chlorophyll-derived sensitizer known as a bacteriochlorophyll C derivative absorbed into the retina itself. This compound extends visual sensitivity into far red wavelengths, allowing the fish to detect the private spectrum it generates. Few known vertebrates possess anything remotely comparable. In effect, the animal modified its own visual chemistry to access a hidden layer of the ocean no competitor could observe. Rows of smaller photophores still line the dragonfish body, producing ordinary blue wavelengths used for counter-illumination camouflage. From below, the fish blends into the faint residual glow filtering downward from the surface, but near the eyes, specialized orbital photophores emit the secret red illumination reserved for hunting. The elegance of the strategy lies not in strength or speed, but in sensory isolation. The loose-jawed dragonfish did not merely improve upon the existing visual ecology of the deep ocean. It abandoned the shared system altogether. Every surrounding creature evolved within one field of perception, while this small predator stepped quietly beyond it into a separate, invisible spectrum where only it could see. A dead whale settling onto the seafloor does not remain hidden for long. Long before scavengers arrive within visible range, before any movement disturbs the sediment around the carcass, chemical traces have already begun spreading outward through the ocean in all directions. Molecules released from decomposing tissue drift into the water column and diffuse slowly through darkness, forming invisible gradients extending across hundreds of meters of empty sea. In the deep ocean, where sunlight never reaches and vision often becomes useless, this chemical landscape forms one of the primary ways life perceives the world. Chemoreception is the detection of dissolved chemical molecules in water, a sensory system comparable to smell and taste combined, though vastly more important in the deep sea than either sense is for most surface animals. Entire ecological systems operate through it. Food, danger, reproduction, territory, and environmental change all leave chemical signatures behind. To deep-sea organisms, the water itself is saturated with information. The sensitivity involved reaches extraordinary scales. Some deep-sea crustaceans can detect organic compounds at concentrations measured in parts per trillion, comparable to identifying a single drop of blood dissolved within an Olympic swimming pool. In an environment where food may appear only rarely, the ability to recognize even the faintest molecular trace becomes essential for survival. The process depends on gradients. A source such as a carcass, living prey animal, or hydrothermal vent releases molecules into surrounding water. Concentrations remain strongest nearest the source and become progressively weaker with distance as diffusion spreads them outward. Chemoreceptive animals navigate by continuously sampling these changing concentrations and adjusting their movements towards stronger signals, effectively climbing an invisible chemical slope through darkness. One of the clearest examples is the hagfish, represented by genera such as Eptatretus and Myxine. These ancient jawless vertebrates have existed with relatively little anatomical change for approximately 300 million years. Their bodies appear primitive beside modern fish, yet they possess one of the most effective scavenging systems in the ocean. A hagfish can detect the chemical traces of a dying or dead animal from remarkable distances across the seafloor, navigating almost entirely through chemoreception rather than vision. When it reaches a carcass, the hagfish often burrows directly into the body through existing openings or softer tissue, feeding from the inside outward while remaining partially protected within the remains themselves. Several hagfish may gather within a single carcass simultaneously, twisting together through the decaying interior in darkness no surface scavenger could tolerate. Its most famous adaptation appears when threatened. Along the hagfish body are specialized slime glands containing tightly coiled protein threads and concentrated mucus compounds. When released into seawater, these threads expand almost instantly into immense volumes of gelatinous slime. A single hagfish can fill a bucket with mucus within seconds. Predatory fish attempting to bite the hagfish often find their gills clogged by the expanding fibers, forcing them to retreat before suffocation occurs. The slime itself has attracted intense scientific interest. The protein threads embedded within it are among the strongest biological fibers known relative to their thickness, comparable in some respects to spider silk. Researchers have studied these fibers for possible applications in surgical materials and lightweight protective fabrics because of their unusual combination of flexibility and tensile strength. Chemoreception also explains how amphipods locate whale falls in the vast darkness of the abyss. As decomposition progresses, amino acids and fatty acids leak steadily into surrounding water, creating elongated chemical corridors stretching outward from the carcass. Amphipods drifting across the seafloor intercept these signals and orient toward increasing concentrations, eventually converging in enormous numbers around a food source they could never have seen. To surface-evolved senses, the deep ocean can appear empty and silent. Yet, beneath that apparent stillness exists another layer of perception entirely, one built not from images or sound, but from dissolved chemistry. Every movement, wound, feeding event reproductive cycle, and geological process releases molecular traces into the surrounding water. The abyss is filled with information continuously exchanged through currents and gradients, a hidden sensory world flowing invisibly through the dark, far below the last traces of sunlight, where food arrives unpredictably and sometimes not at all. The shape of a mouth can determine whether an animal survives another month or disappears into the dark unnoticed. In these depths, evolution repeatedly favors expansion, flexibility, and excess. Jaws widen beyond proportion, bones loosen, connective tissue softens, and stomachs either become grotesquely elastic or fade into something almost unrecognizable. The deep sea does not reward restraint. It rewards anything capable of extracting the maximum possible energy from the rare moment when prey finally appears. Several deep-sea fish families, particularly the Saccharopharyngidae and Eurypharyngidae, evolved digestive systems so specialized that conventional stomach anatomy began to disappear entirely. Others moved toward the opposite extreme, developing expandable internal cavities capable of engulfing prey far larger than themselves. Surface ecosystems, where food appears frequently and competition is constant, rarely produce such extremes. In the abyss, scarcity reshapes anatomy with unusual freedom. Among the strangest examples is the pelican eel, Eurypharynx pelicanoides. Its body appears thin and whip-like for most of its length, yet attached to the front is an enormous hinged mouth capable of opening wider than the rest of the fish itself. The jaws are loosely connected, allowing them to spread outward into a vast pouch resembling the throat of a pelican, which inspired the animal's common name. Although the fish usually measures around 75 centimeters long, much of that length belongs to its tapering tail rather than its central body. The tail itself ends in a small bioluminescent organ, glowing pink-red in darkness. Researchers believe this luminous tip may function as a lure, attracting curious animals toward the waiting mouth. In an environment where movement is costly and prey unpredictable, the ability to draw food inward rather than chase it becomes deeply valuable. The digestive anatomy of the pelican eel reflects the same principle. Its stomach stretches dramatically to accommodate prey larger than would seem physically possible for such a narrow fish. Tissue elasticity replaces muscular strength. The animal survives not by consuming food often, but by exploiting each successful capture to the fullest extent possible. Other species refined the same strategy differently. The whipnose anglerfish, belonging to the genus Gigantactus, transformed its luminous lure into an extraordinary extension projecting far beyond the body itself. Instead of positioning the glowing esca directly above the head like most anglerfish, Gigantactus suspends the lure from a filament that may extend several times the fish's own body length into surrounding darkness. This arrangement likely reduces the risk of alerting prey to the predator's presence. Small animals investigating the drifting light encounter the lure first, while the anglerfish remains hidden farther away in darkness, almost disconnected from its own bait. The entire body becomes secondary to the feeding apparatus itself. The stoplight loosejaw, Photostomias gurnei, combines another variation of these deep-sea feeding strategies. Like the dragonfish discussed earlier, it produces red bioluminescence invisible to most surrounding animals. Its jaws open to consume prey approaching half its own body size, an extraordinary ratio for a relatively small predator. Flexibility matters more than precision. Any available calorie becomes worth capturing. In the viperfish, Choliotis sloani, the problem took on a more mechanical form. Its fangs grew so long they no longer fit inside a closed mouth. Instead, the teeth curve outward along the exterior of the skull when the jaws shut. During a strike, the fish lunges upward at high speed, impaling prey on these oversized fangs before closing its jaws around the stunned animal. The teeth function almost like trapping spikes before becoming chewing structures. All of these anatomies emerge from the same environmental pressure. Food encounters in the deep sea may occur weeks or months apart. A predator limited to consuming only small prey risks wasting precious opportunities. Evolution, therefore, pushes mouths wider, stomachs farther, connective tissue looser, and feeding mechanisms toward extremes rarely tolerated in surface ecosystems. These fish did not abandon the logic of biology. They extended one of its oldest principles to an extraordinary conclusion. In the abyss, survival belongs not to the fastest hunter or the strongest swimmer, but to the animal capable of turning a single rare encounter into the greatest possible return of energy before darkness closes again. Across the abyssal plains, where the sea floor stretches outward in enormous fields of gray sediment, life often appears reduced to the smallest possible scale. Tiny crustaceans drift between grains of mud, bacterial films spread across the surface in thin, invisible layers, and organic particles settle slowly from the water above like dust sinking through still air. Yet among these quiet landscapes, live organisms so strange that, at first glance, they seem impossible to classify correctly. They resemble clusters of coral, collapsed sponges, or fragile stone lace scattered across the sediment. In reality, each one is a single living cell. These organisms are xenophyophores, enormous members of the foraminifera, a group of amoeba-like life forms known for building protective shells called tests. Unlike animals, plants, or fungi, a xenophyophore contains no organs, no tissues, and no division into specialized body parts. Every process required for survival, respiration, feeding, waste removal, reproduction, and repair occurs within the boundaries of one continuous cell membrane. The largest known species, Syringamina fragilissima, reaches nearly 20 centimeters in diameter, making it the largest confirmed individual cell on Earth. At that scale, the organism becomes visible to the naked eye as a branching, delicate structure resting across the sediment like brittle underwater roots. Some resemble tangled webs, while others spread outward in fan-shaped patterns or irregular spirals. Their appearance changes depending on the material available around them. Xenophyophores build their tests from particles gathered directly from the seafloor. Mineral grains, fragments of shell, sponge, spicules, and other microscopic debris are collected and bound together with a protein cement secreted by the cell itself. The result is a fragile external framework that provides both support and surface area. Unlike the hard skeletons of animals, these structures remain delicate enough to crumble easily when disturbed by currents or submersible contact. They inhabit some of the deepest places on Earth, living primarily across abyssal plains between 500 and 10,600 meters deep. Researchers have documented xenophyophores at the bottom of the Mariana Trench itself, existing under pressures capable of crushing most known life. In these depths, where food arrives only in sparse drifting particles, their unusual body plan becomes surprisingly effective. Feeding occurs through pseudopods, temporary extensions of the cell body that spread outward into surrounding sediment. These thin flowing structures gather bacteria and organic particles before retracting them inward. The process resembles a constantly shifting web extending across the mud. There are no jaws, no stomach chambers, and no circulatory system carrying nutrients between tissues. Everything happens within the chemistry of one enormous cell. Their importance extends far beyond their own survival. Abyssal plains are often biologically monotonous environments dominated by soft, unstable sediment with few solid surfaces available for attachment. Xenophyophore tests provide rare, hard substrate in this otherwise featureless terrain. Worms settle inside their branching cavities, small crustaceans shelter among the openings, and juvenile echinoderms anchor themselves against the framework. Entire micro-communities form around structures created by a single cell. Another mystery lies hidden inside the organism itself. Xenophyophores accumulate unusually high concentrations of barium sulfate crystals, known as barite, within their bodies. These concentrations far exceed anything found in surrounding sediment. Scientists still do not fully understand why the organisms gather these crystals or what biological purpose they serve. Over geological time, their barite-rich waste contributes measurably to the chemistry of abyssal sediments, quietly influencing the composition of the deep seafloor itself. What makes xenophyophores especially remarkable is the problem they present to biology at the cellular level. Most cells remain microscopic because molecular transport becomes inefficient across large distances. Nutrients, oxygen, and waste products cannot easily move through enormous volumes of cytoplasm quickly enough to sustain life. Yet xenophyophores pushed far beyond those presumed limits. At nearly 20 centimeters across, they transport material internally over distances unmatched by any other known single cell. In the stillness of the abyss, evolution arrived at a solution so simple it almost disappears into abstraction. No organs were added, no tissues evolved, and no greater complexity emerged. One cell simply continued growing, adapting the ancient blueprint of single-celled life until it became large enough to alter the architecture of the seafloor around it. Long before human beings lowered instruments into the abyss, the deep ocean was already carrying voices across planetary distances. A single sound released into the right layer of water could travel farther than mountains, farther than coastlines, farther than entire nations. In darkness, where vision collapses after only a short distance, the ocean became a world organized not by sight, but by vibration. Pressure, temperature, and water density shaped invisible corridors through which sound could move almost endlessly, bending gently through the deep like light trapped inside glass. Sound behaves differently underwater than it does in air. In seawater, it travels at roughly 1,500 meters per second, approximately 4.3 times faster than through the atmosphere above. Molecules in water sit closer together than those in air, allowing vibrations to pass rapidly between them. In the deep ocean, where currents move slowly and temperatures remain stable, those vibrations can persist for astonishing distances without fading completely away. Between roughly 600 and 1,200 meters depth lies one of the ocean's strangest physical structures, the SOFAR channel, short for sound fixing and ranging channel. At this depth, temperature and pressure interact in a way that creates a minimum speed zone for sound. Waves drifting upward encounter warmer water where sound speeds increase, bending them downward again. Waves drifting deeper encounter rising pressure, which also increases sound speed, curving them back upward. The result is an acoustic corridor that traps sound inside itself. Discovered during the 1940s by American oceanographer Maurice Ewing and his colleagues, the SOFAR channel quickly became important during the Second World War. The United States Navy developed the SOSUS network, the sound surveillance system, which used underwater hydrophones to monitor submarine movement across entire ocean basins. A sound released thousands of kilometers away could still arrive detectable inside the channel. Underwater landslides, volcanic eruptions, and even the 1961 nuclear test, known as Project Fishbowl, produced signals that traveled enormous distances through this hidden acoustic layer. Whales evolved within this environment long before human technology recognized it. The blue whale, Balanoptera musculus, produces calls between 10 and 40 hertz, frequencies so low that humans perceive them more as vibration than sound. These calls can reach approximately 188 decibels, making them the loudest known sounds produced by any animal. Released into the SOFAR channel, they can travel across thousands of kilometers of ocean water. Somewhere in that enormous acoustic world moves a whale unlike any other yet identified. Since 1989, hydrophones across the Pacific Ocean have repeatedly recorded a solitary whale call at 52 hertz, a frequency outside the normal range of known whale species. The animal migrates in patterns consistent with whale behavior and returns season after season, yet no visual identification has ever confirmed what species it belongs to. Its calls continue moving through the ocean alone, distinct from every surrounding voice. Deeper still, another giant produces sounds even more powerful. Sperm whales generate echolocation clicks reaching approximately 230 decibels, among the loudest biological sounds ever measured. These clicks originate within the spermaceti organ, occupying much of the whale's immense forehead. The sound waves sweep outward through darkness, mapping squid and fish through reflected echoes. Some researchers believe the intensity may even stun prey at close range before capture. For millions of years, these acoustic systems shaped life beneath the surface, with little interruption. Yet over recent decades, another sound entered the ocean in growing volume. Commercial shipping produces continuous low-frequency noise in nearly the same frequency bands used by large whales. Engine vibration, propeller cavitation, and industrial traffic spread across major shipping corridors without pause. Acoustic studies have shown that ambient ocean noise in busy regions has increased by roughly 3 decibels per decade since the 1960s. Though numerically small, that increase effectively halves the distance over which whales can communicate. Calls that once crossed enormous stretches of ocean now dissolve into mechanical interference long before reaching distant listeners. Even in the deepest waters, where sunlight never arrives and pressure reshapes biology itself, the ocean remains filled with conversation. It travels through invisible corridors older than humanity, carried by physics through darkness vast enough to hide entire worlds between one voice and another. Each evening, while coastlines darken and surface waters begin reflecting the first faint colors of night, an immense movement begins beneath the ocean that almost no human being ever sees. Across every major sea on Earth, countless animals rise slowly upward through the water column from depths where daylight barely exists. They travel in such numbers that sonar instruments often mistake them for the seafloor itself. Then, before dawn arrives, they sink again into darkness, carrying the remains of the surface world down with them. This movement unfolds within the mesopelagic zone, more commonly called the twilight zone, extending from roughly 200 to 1,000 meters below the ocean surface. At its upper boundary, less than 1% of sunlight remains. At its lower boundary, light disappears entirely. The water exists in a suspended state between visibility and complete darkness, where outlines fade gradually rather than vanish all at once. Here, every day and night, trillions of animals participate in the largest migration on Earth by biomass. Unlike the migrations of wildebeest across savannas or birds across continents. This migration happens continuously, every 24 hours, across nearly every ocean basin simultaneously. Lanternfish of the family Myctophidae rise upward through the dark alongside krill, copepods, medusae, arrowworms, and the extraordinarily abundant bristlemouth fish of the family Gonostomatidae. Bristlemouths alone may represent the most numerous vertebrates on Earth. Many are small enough to fit comfortably within a human hand, yet together they form an almost unimaginable living mass spread through the twilight zone. The rhythm guiding this migration is ancient and precise. During daylight hours, these animals remain deeper in darkness, where visual predators struggle to detect them. Tuna, dolphins, seabirds, and larger predatory fish dominate the illuminated surface waters during the day. Remaining hidden below dramatically increases survival. Yet the surface contains something the depths cannot provide. Phytoplankton, microscopic drifting organisms powered by sunlight, form the foundation of most marine food chains. They remain concentrated in the sunlit upper ocean where photosynthesis occurs. At night, when darkness spreads across the surface and visual predation weakens, twilight zone animals ascend to feed. The migration resembles an enormous breathing cycle within the ocean itself. Billions upon billions of organisms rise toward the surface after sunset, consume food through the night, then descend again before sunrise. Sonar recordings often reveal dense biological layers, lifting upward through the water in synchronized waves, spanning entire regions of ocean. This movement quietly influences the chemistry of the planet. After feeding near the surface, migrating animals carry carbon-rich material downward inside their bodies. As they descend, they respire, excrete waste, and eventually die at depth, transporting surface carbon into the deep ocean. Scientists estimate this active biological transport contributes an additional 15 to 40% beyond the passive marine snow process already discussed earlier. Among the most important participants are lanternfish. Though rarely seen directly by humans, global estimates suggest mesopelagic fish biomass may range between 1 and 10 billion tons, potentially exceeding the combined mass of all commercially harvested fisheries on Earth. Entire continents of living fish drift nightly through darkness, almost unnoticed. Lanternfish communicate and recognize one another using intricate arrangements of photophores, specialized light-producing organs distributed across their bodies. Each species possesses its own distinct luminous pattern, functioning almost like a biological fingerprint. In darkness, where shapes disappear quickly, these constellations of blue light allow schooling behavior and species recognition without revealing the animals too clearly to predators below. Despite its enormous ecological importance, the twilight zone remains among the least studied environments in the ocean. It exists in an awkward depth range, too deep for many traditional fishing methods and scientific sampling tools, yet too shallow to attract much of the expensive, deep submersible exploration directed toward abyssal trenches and hydrothermal vents. As a result, one of the largest ecosystems on Earth continues moving mostly unseen each night beneath the surface. Entire populations rise and descend through darkness in synchronized silence, carrying carbon, energy, and light between worlds of sun and abyss, with a precision refined over millions of years. Far beneath the ice of certain distant moons, there may be oceans that have never seen sunlight even once in the history of the solar system. No sunrise has ever crossed them, no atmosphere stirs above them, And yet modern astrobiology increasingly treats these hidden oceans not as impossible places for life, but as some of the most promising environments beyond Earth. That shift in scientific thinking began only after humanity discovered what already existed in the deep sea below its own world. For much of modern history, scientists assumed sunlight formed the absolute foundation of all complex ecosystems. Then, hydrothermal vents were discovered along the ocean floor, surrounded by living communities powered not by solar energy, but by chemistry itself. Sulfides, methane, mineral gradients, and geothermal heat replaced sunlight entirely. Life proved capable of surviving through pathways far older and stranger than surface biology had suggested. That realization transformed the search for extraterrestrial life almost immediately. Among the most important targets is Europa, one of Jupiter's large moons. Beneath Europa's frozen outer shell, estimated to range between 10 and 30 kilometers thick, researchers believe an immense liquid ocean exists. Tidal forces generated by Jupiter's gravity continuously flex the moon's interior, creating frictional heat that prevents the ocean from freezing solid. In many ways, the environment resembles Earth's deep ocean beneath polar ice, isolated from sunlight, but potentially rich in chemical energy emerging from interactions between water and rock far below. Saturn's moon Enceladus deepened that possibility even further. In 2005, observations from the Cassini spacecraft confirmed enormous plumes erupting through fractures near the moon's southern pole. These plumes contained water vapor, microscopic ice grains, hydrogen, silica nanoparticles, and carbon-based molecules. The chemistry strongly suggested active hydrothermal systems beneath the surface ocean, remarkably similar to vent systems found on Earth's seafloor. The deep ocean had already shown that hydrothermal environments can support entire ecosystems independent of sunlight. Suddenly, icy moons no longer appeared sterile by default. Organisms, called piezophiles, became especially important within this discussion. These pressure-loving microorganisms inhabit Earth's deepest trenches, functioning under crushing conditions once considered biologically impossible. Their existence demonstrated that cellular machinery can remain stable and active under pressures comparable to those expected within subsurface extraterrestrial oceans. Rather than speculative theory, they became living proof that life's tolerances extend far beyond earlier assumptions. Then there are tardigrades, microscopic animals often called water bears, found in habitats ranging from moss to deep-sea sediments. Their resilience stretches almost beyond intuition. Tardigrades survive temperatures approaching -272 degrees Celsius, heat exceeding 150 degrees Celsius, radiation levels that would destroy most multicellular life, and pressures many times greater than those found within the Mariana Trench. Some have even survived direct exposure to outer space in low Earth orbit during experiments conducted by space agencies. Their survival reopened scientific discussion around panspermia, the hypothesis that microbial life might travel naturally between planets inside rocks blasted free by asteroid impacts. If certain organisms can endure vacuum, radiation, and prolonged dormancy, then interplanetary transfer no longer appears entirely beyond biological possibility. At the same time, Earth's deep sea may also preserve clues not merely about life elsewhere, but about how life itself first emerged here. Alkaline hydrothermal systems, such as the Lost City vent field, discovered in 2000 at depths between 700 and 800 meters along the mid-Atlantic ridge, have become central to origin of life research. Unlike the acidic black smokers discussed earlier, Lost City releases alkaline fluids and builds immense calcium carbonate towers, rising as high as 60 meters from the seafloor. Laboratory studies suggest environments like these may provide the chemical gradients capable of driving the formation of RNA, the molecule widely believed to have preceded DNA as life's earliest genetic system. In this way, the deep ocean became more than an isolated ecosystem. It became both archive and mirror, preserving conditions similar to Earth's earliest biological beginnings, while also offering a model for oceans hidden beneath ice on worlds orbiting far beyond the horizon of this one. Even now, after centuries of navigation, satellite imaging, robotic submersibles, and global scientific collaboration, the largest habitat on Earth remains mostly undescribed. The deep ocean covers more of this planet than every continent combined, yet marine biologists estimate that fewer than 10% of its species have been formally identified and named. Conservative estimates suggest somewhere between 500,000 and 10 million deep-sea species may still remain undiscovered, drifting through trenches, burrowing beneath sediment, or attached to volcanic stone in darkness no human eye has witnessed directly. The scale of that absence changes the meaning of exploration itself. On land, most large animals have been cataloged for generations. In the deep sea, entire ecosystems continue to appear unexpectedly during ordinary surveys. The Seabed 2030 project, a global effort led by the Nippon Foundation and GEBCO to map the entire ocean floor, has repeatedly demonstrated this reality. Expeditions conducted between 2022 and 2025 consistently returned not only with bathymetric measurements, but with biological discoveries as well. New tube worms, unfamiliar fish, translucent crustaceans, and previously unknown invertebrates emerged from regions located less than 100 kilometers from places scientists had already surveyed before. Vast portions of the seafloor remain so sparsely explored that neighboring valleys can contain communities separated for evolutionary timescales longer than human civilization itself. Beneath even those ecosystems lies another world, still harder to measure. Within sediments buried below the seafloor, archaea and bacteria form a deep, subsurface biosphere extending kilometers beneath the ocean bottom. A study published in 2018 estimated that this hidden microbial realm may contain as much carbon as all plant life on Earth's surface combined. These organisms survive in darkness under enormous pressure, metabolizing at rates so slow that individual cells may divide only once over spans measured in centuries. Their genetics represent one of the least cataloged reservoirs of biological information on the planet. At the same time, many of the most basic interactions between larger deep-sea animals remain poorly understood. Chemical communication, the invisible language carried through seawater, has only been studied in a handful of species. The exact molecules used to signal danger, identify mates, mark territory, or coordinate reproduction are still mostly unknown. Entire sensory worlds operate continuously beyond human perception, composed of gradients and compounds drifting through black water with no visible trace. Meanwhile, the ocean itself is changing faster than many deep ecosystems may be capable of adapting to. As surface temperatures rise, the thermocline separating warm upper water from colder depths is strengthening and deepening. This reduces vertical mixing, slowing the transport of oxygen and nutrients into midwater ecosystems. Scientists understand the physics of this process, yet the biological consequences remain difficult to model with confidence. The twilight zone migrations that regulate planetary carbon movement may shift in ways not yet predictable. Another uncertainty arrives from the sea floor itself. Across abyssal plains lie immense fields of polymetallic nodules, slow-growing accumulations of manganese, cobalt, nickel, and copper resting on sediment that has remained undisturbed for millions of years. Commercial interest in deep-sea mining continues to expand. Simulations suggest that sediment plumes generated by mining machinery could spread across areas thousands of times larger than the excavation site itself, burying delicate organisms adapted to extraordinary environmental stability. Recovery times for abyssal ecosystems are estimated not in years, but in centuries. Even intelligence remains an unanswered question. Cephalopods living in shallow seas have demonstrated memory, problem-solving, tool use, and behavioral complexity unlike almost any other invertebrates. Yet the cognition of deep-sea cephalopods, including colossal squid and other rarely observed species, remains almost entirely unstudied. And, somewhere within the ocean's vast acoustic darkness, the unidentified whale, calling at 52 Hz, continues its migrations decade after decade, recorded since 1989, yet still unseen, still unnamed, still unresolved, within the immense, unfinished map of the sea. Long before human beings built cities, named constellations, or understood what an ocean truly was, life already existed in darkness beneath kilometers of water. It fed without sunlight, survived under pressures capable of crushing steel, and evolved forms so unfamiliar that even now they appear almost imagined when first seen through submersible cameras. The creatures described throughout this journey did not arrive through randomness or excess. Every transparent skull, every chemical metabolism, every impossible jaw or pressure-resistant cell membrane exists because countless other forms failed before them. Biology leaves little room for arbitrary design. What appears strange from the surface is often simply the visible shape of survival under conditions most organisms cannot endure. The anglerfish fused permanently to its mate represents one solution to a world where encounters are unimaginably rare. The snailfish, with its gelatinous body, reflects the chemistry required to survive at trench pressure. The Pompeii worm endures beside water hot enough to unravel ordinary proteins because lineages unable to tolerate such heat disappeared long ago. Evolution did not invent these forms to surprise human observers. It produced them because, in those environments, they worked. For much of scientific history, life itself was defined more narrowly than it is today. Before the discovery of hydrothermal vent ecosystems in 1977, most working assumptions about biology treated sunlight as the ultimate foundation of complex life on Earth. Oceans, forests, grasslands, and coral reefs all ultimately traced their energy back to the sunday. Then scientists descended into the deep Pacific and found ecosystems thriving entirely on chemical energy emerging from the seafloor. That moment changed more than marine biology. It changed the boundaries of what life was understood to be capable of becoming. Since then, the revision has continued expanding. Organisms have been found surviving temperatures once considered lethal to complex cells, pressures once believed incompatible with vertebrate biology, and oxygen levels too low for ordinary metabolism. Each discovery quietly altered the map of possibility. There is also a deeper lesson carried through the history of deep-sea exploration itself. Again and again, researchers lowered cameras, nets, and instruments into darkness, expecting one thing and retrieving another. The pattern repeats across generations. The transparent dome of the barrel-eye fish remained misunderstood because specimens collapsed before reaching the surface. Giant tube worms contradicted assumptions about ecosystems without sunlight. Cold seep communities emerged where no volcanic heat existed. Entire biological strategies appeared that no theoretical model had predicted beforehand. The deep ocean repeatedly demonstrated that unexplored environments resist confident assumptions. At the same time, the organisms themselves possess no awareness of the impossibilities humans assign to them. The tardigrade surviving vacuum does not experience itself as extraordinary. The vampire squid, drifting through oxygen minimum zones, has no concept of evolutionary rarity. A tube worm growing meters tall without a mouth or digestive tract carries no understanding that surface biology once considered such a thing impossible. The category of impossible exists almost entirely in the human mind, built from the limits of previous observation. And perhaps the deepest reversal of perspective comes from time itself. Earth is approximately 4.5 billion years old. Complex surface ecosystems, forests, mammals, birds, flowering plants, and human civilizations occupy only a comparatively recent fraction of that span. For most of Earth's history, life was microbial, aquatic, and hidden beneath the surface. The bright terrestrial biosphere humans experience every day may not represent the standard condition of life on this planet at all. It may be the exception. The deep, cold, pressurized ocean may be closer to life's original and most enduring state. We often speak as though the age of biological discovery belongs to the past, to the great voyages of classification, to expeditions like the HMS Challenger in the 19th century. Yet deep-sea biology remains an active frontier in the purest sense. Many of its most significant discoveries have occurred only within the last 50 years, and each new descent still carries the possibility of encountering forms of life never before observed by human eyes. Long before forests spread across continents, before birds crossed the sky, before insects hummed through warm air or mammals walked across grasslands, the deep ocean was already alive. In darkness, beneath ancient seas, organisms were evolving, competing, consuming one another, reproducing, and vanishing into extinction without ever sensing the existence of the world above them. The abyss predates nearly everything human beings instinctively associate with life on Earth. It existed before trees, before flowers, before bones hardened into the shapes of familiar animals. Even now, after all human exploration, it continues almost entirely indifferent to the surface. A snailfish, drifting more than 8,000 meters below sea level, has never experienced sunlight. No storm has disturbed its world. No season changes the water surrounding it. Ice ages came and went above, while the trench remained cold and black. The ancestors of those fish were already living in darkness, while the earliest hominids were still learning to walk upright across African plains. Entire civilizations rose, disappeared, and became archaeology without altering the stillness below. At this very moment, across waters covering most of the planet, bioluminescent signals continue flashing silently through the dark. Predators illuminate prey invisible to other eyes. Lanternfish pulse species specific patterns to remain within their schools. Tiny crustaceans erupt in blue light when disturbed. Dragonfish sweep private beams of red through the water where almost nothing else can see. The ocean contains an uninterrupted conversation of living light happening continuously across depths no human observer will ever personally visit. And, It has been happening for immense spans of evolutionary time. Somewhere in ancient seas, hundreds of millions of years ago, long before forests covered land, organisms first began producing light through chemistry. Those signals traveled through black water before complex eyes had fully evolved to perceive them clearly. The deep ocean became luminous long before humanity arrived to notice it. Far below the surface, stillness dominates almost everything. At depths around 4,000 meters, motion itself becomes subtle. No wind reaches there. Surface waves vanish far above. Day and night lose meaning entirely. There are no sunrises, no sunsets, no weather systems crossing the sky. Marine snow drifts downward slowly through near-freezing water, carrying the remains of distant surface ecosystems into darkness. Occasionally, a pulse of bioluminescence appears and disappears again, brief as a thought. Within that stillness, life became extraordinary. The creatures encountered throughout this journey reveal something larger than unusual anatomy or unfamiliar ecosystems. They reveal that biology's apparent rules are not rigid walls, but provisional boundaries shaped by incomplete observation. Again and again, the deep ocean expanded those boundaries. Life without sunlight. Animals thriving beside near boiling water. Organisms surviving pressures capable of crushing submarines. Single cells visible without microscopes. Predators hunting with invisible wavelengths of light. Entire ecosystems fueled by chemistry emerging from rock. The most alien environments humans can imagine on distant worlds already exist here on Earth, hidden beneath oceans that most people will never see directly. And even now, the exploration remains unfinished. Somewhere below the ship lanes and satellite routes, below fishing nets and research vessels, below every location visited by human divers, there are organisms alive tonight that have never been seen. Some possess no scientific name. Some belong to lineages unknown to biology. Others may carry solutions to environmental pressures humans have not yet encountered, adaptations to problems science has not even recognized as possible. In water so cold, dark, and pressurized that it resembles outer space more than any familiar landscape, they continue living exactly as their ancestors did long before humanity appeared. They drift through trenches, crawl across mineral plains, pulse with hidden light, and follow chemical trails through black water that has never reflected the sky. The ocean does not know we exist, and for hundreds of millions of years, it never needed to.